Hey, before we start the video, if you like this sort of content and you want more, come check out Med School Bootcamp. We have content for Step 1, Step 2, Anatomy, and OMM. Right now, we're about to watch the Biochemistry section. And if you're looking for downloadable PDFs, along with shorter videos and quizzes after each video to help you reinforce the information, then you can find it all on Med School Bootcamp. We also have a Step 1 question bank with some of the most representative questions to help you prepare for Step 1. You can also create a custom Step 1 study schedule when it's time for dedicated. So if you're looking to crush it in med school this year and pass Step 1, come check out Med School Bootcamp. It's going to be your first link down in the description. Now on to the video.
Hi everyone, my name is Paige and thank you so much for joining me for our chapter on lipid metabolism. We have a ton to cover in this chapter. We're going to start off in this video with our introduction to lipids, refreshing ourselves on some of the structures and functions of different lipid types. And then in the next couple videos, we'll talk about cholesterol and triglyceride, absorption and digestion, and transport. We'll do a pharm lesson on all of the lipid control drugs we've got, move into a group of conditions called familial dyslipidemias. We'll talk about what goes on with lipids in the periphery in adipocytes with lipogenesis and lipolysis. Then we'll cover beta oxidation, which is how we degrade our fatty acids into energy. We'll talk about de novo fatty acid synthesis, some disorders of fatty acid metabolism. We will finally have our formal discussion on ketones, and we'll wrap everything up with a summary video about the fed versus the fasting state. So let's kick off our discussion with an introduction to lipids. So lipids are non-polar organic molecules, and there are lots of larger structures that lipids can take, but the building block is going to be called a fatty acid. And those fatty acids can come together and esterify, form an ester, and assemble into larger structures like triglycerides, steroids, and structural lipids. So let's start off by talking about fatty acids, those building blocks. The structure of a fatty acid is a carboxylic acid with a linear carbon chain. And if you blocked out all your Gen Chem from undergrad, which I don't blame you for, carboxylic acid, right, is this right here, C double bond O OH. And then from there, it's just attached to this long carbon chain of variable lengths. We can classify fatty acids based on the length of that chain. So a short chain fatty acid is going to have 1 to 6 carbons. And that's starting from that first carbon right here in the middle of that carboxylic acid. And you count onwards from there. Medium chain has 7 to 12 carbons. Long chain fatty acid has 13 to 20 carbons. A very long chain fatty acid is going to have more than 20 carbons. And then throughout the chapter, you'll hear us refer to even chain versus odd chain carbons. Again, just referring to whether or not there is an even number or an odd number of carbons along that chain. There's some special implications of odd chain fatty acids versus even chain when we talk about beta oxidation. Another way we can classify fatty acids is their saturation level. So we can have saturated or unsaturated fatty acids. A saturated fat does not have any double bonds, whereas an unsaturated fat can be mono or polyunsaturated. That's just referring to the number of double bonds, and this can be an a cis conformation or a trans conformation. Let's take a look at the visual to explain that a little more. Up top here, we've got a saturated fat. In the bottom here, we have an unsaturated fat. You can see a saturated fat has unique attachments at every single possible bond site. Compare that to an unsaturated fat. You can see right here is the double bond, and because we have 2 bonds connecting these carbons, those are not unique attachments. That second bond is taking the place of what could be 2 more hydrogen bonds. So we refer to that as an unsaturated fat. This is monounsaturated, but if we had another double bond, we would call that polyunsaturated. And then we mentioned cis versus trans fats. This is an example of a cis fat. What that means is this double bond right here creates a kink in the chain. If the 2 legs in the kink of the chain are pointing towards the same direction, that's a cis fat, right? It kind of looks like a little boat. However, if we had a chain like this, and then we had a double bond, and then the next leg after the double bond pointed in the opposite direction, creating more of a Z here, as opposed to the sort of boat configuration of the cis fat. When they point in the opposite directions and create the Z, that's called a trans fat. And as you can see, where these cis fats create a giant kink in the chain here, those trans fats actually stay pretty in line with the original chain. Those trans fats don't deform that chain in the same drastic way that a cis fat would. So saturated fats, in addition to trans unsaturated fats, can pack together really tightly. You can see how a lot of those chains would be able to just stack right up on top of each other, whether you had saturated fats or trans unsaturated fats. They pack together really tightly in membranes, and that decreases membrane fluidity, which increases the risk of cardiovascular disease. But we have even more fatty acid nomenclature to cover, and that is the omega nomenclature. Omega nomenclature applies to unsaturated fatty acids, so fatty acids that have a double bond in them. What you're going to do is you're going to count the carbons from the methyl end. They call it the omega end. That's the end farthest away from the carboxylic acid. Then you're going to count the distance between that omega end and the first carbon of the first double bond you encounter. So let's zoom in down here for a bit of an example. So we're coming up on triglycerides next. This is just an example of a triglyceride. Got a glycerol backbone. We're just here focused on these fatty acid tails for now to teach you about omegas. Right here is the carboxylic acid. So the opposite end from that carboxylic acid is called the omega end, right? That's the end with that methyl group sticking off the end. So here's our omega end. And then to figure out what the omega number is, we're going to count backwards from the omega. So here's 1, 2, 3. And then you can see at that third carbon from the omega end is the first carbon involved in the first double bond we encountered. That means that this fatty acid tail right here is an omega-3 fatty acid. Now you're likely to encounter further specificity with the omega nomenclature. For example, this fatty acid here on the bottom, because it has 18 carbons and 3 double bonds, would be called an 18-3 omega-3 fatty acid. Now we'll talk about this when we talk about fatty acid synthesis, but a lot of these polyunsaturated fatty acids cannot be synthesized within the body. They have to be consumed. It's difficult for your body to add these double bonds into these chains that are very, very long. So a lot of omega-3 and omega-6 fatty acids are essential fatty acids, meaning they must be taken in from the diet. Some examples of omega-3s that you might encounter are alpha-linolenic acid, which is actually the structure of this fatty acid tail that we were talking about right here, icosapentaenoic acid, and docosahexaenoic acid. And omega-3s are very important because they are anti-inflammatory. The way that they're anti-inflammatory is they will decrease the arachidonic acid in platelet membranes. That causes decreased TXA or thromboxane production, which decreases platelet aggregation and decreases cardiovascular disease, and that's why they're anti-inflammatory. Now, on the opposite end of the spectrum from omega-3s, we have omega-6s, and omega-6s are pro-inflammatory. Some examples of omega-6s are linoleic acid. Don't confuse linoleic acid with alpha-linolenic acid. Linoleic acid is the omega-6. It increases arachidonic acid, which increases prostaglandins and leukotrienes, which are inflammatory mediators. So now that we understand fatty acids and all of their nomenclature, we can understand some of the larger lipids made up from these fatty acids. The first is triglycerides, and this is the storage form of fats and fatty acids. These triglycerides are going to be synthesized in the liver and the adipose tissue, right, or fat cells. And the composition of a triglyceride is 3 fatty acids plus 1 glycerol linker, and they're linked by an ester bond. So we were using this triglyceride to look at sort of its individual fatty acid tails, but as you can see here, it is actually a triglyceride. So here is our glycerol backbone, and then here you've got your ester bonds linking that glycerol backbone to that fatty acid tail. And from this storage form, fatty acids can be liberated and broken down via beta oxidation for energy. Next, we'll talk about steroids, and steroids look a little bit different from triglycerides. These are going to have a bunch of rings. So we've got 3 6-sided and 1 5-carbon ring. So here is an example of cholesterol. You can see 6-carbon ring, 6-carbon ring, 6-carbon ring, 5-carbon ring, and then a tail coming off the end here. So you can see this cholesterol is a really big, bulky molecule, going to help space out some of those other more tightly packed elements of cell membranes. And so it is going to increase cell membrane fluidity. Cholesterol is also a precursor for a ton of things throughout your body. It's very important, and we're going to talk in detail about cholesterol in this chapter. It's a precursor for hormones, for bile acids, for vitamins, and it can be absorbed or it can be synthesized de novo. So involved in a lot of different pathways. One term you're going to come across as we move throughout this chapter is the term cholesterol ester. A cholesterol ester is a dietary lipid, and it's just a fancy word for cholesterol with an extra long fatty acid tail attached to it. So here's our cholesterol ester, where you've got our cholesterol just like you see it in the image above, but it is ester bonded via this ester bond right here to an extra fatty acid tail. That makes it a cholesterol ester. And then finally, we'll talk about structural lipids, and this is a class of lipids that includes things like phospholipids, sphingolipids, and glycolipids. I don't have a picture here for you because there's a ton of different structures within these categories. You can look up some pictures for inspiration, but in essence, what you need to know is these are cell membrane components. They're involved in structure, signaling, and they have receptor function. So let's wrap it up. In this chapter, we are talking about lipids, a major energy source in the body. We talked about fatty acid building blocks and how they can be used to build up larger molecules like triglycerides, steroids, and structural lipids. We talked about some of the nomenclature with fatty acids, right? By chain length, by saturation status, and then within unsaturated fatty acids, we talked about that omega nomenclature, including our anti-inflammatory omega-3s and our pro-inflammatory omega-6s. We talked about how we can make a lot of these lipids or how we can take them in from our diet. And that dietary, digestion, and absorption is what we're going to be talking about in the next video. Till then, stay tuned. That's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on lipid digestion and absorption. So now that we've reviewed our fundamentals of lipids, let's talk about how we actually get them from outside in our food into our bloodstream. And the actual absorption of lipids from our gut into our blood is not super straightforward, and that has to do with their structures right here. If you take a look, both triglycerides and these cholesterol esters both are these big, bulky molecules. They've got these long hydrophobic tails, and that makes them insoluble, and their size makes it difficult for them to cross through all the membranes they need to cross through in the absorption pathway. So in this video, we're going to learn how our body cleaves apart these big molecules, pushes them across membranes, and then reassembles them on the other side of the membrane for packaging. So the way to really think about this is kind of like this IKEA bookcase I got behind me, right? When I moved in here, that bookcase was too big to fit through the door. So what did I do? I took it all apart, I brought all the pieces through the door, and then I reassembled the bookcase on the other side. Your body's gonna do the same thing with these big bulky molecules right here. It's gonna take them apart, bring all the pieces across the membranes, and then reassemble them on the other side. So our overview image for the absorption of both triglycerides and cholesterol is over here on the right, but we're actually gonna start by talking about the outline, and then we'll pull it all together on this image sort of at the end. Let's focus on triglycerides and cholesterol sort of separately in turn. We'll start by talking about triglycerides. So when we consume a big fatty meal full of triglycerides, enzymes called lipases are going to cleave these triglycerides down into their components. And those components are either going to be 2 fatty acids and a monoglyceride, or 3 fatty acids and a glycerol molecule. So if you take a look at the structure of a triglyceride right here, you've got the glycerol molecule kind of right here, and then these 3 long fatty acid tails that are coming off. So if you want to break this up into pieces, you can break it into a monoglyceride like this, that glycerol backbone plus 1 fatty acid along with 2 fatty acid tails right here. Or alternatively, you could cleave off all of those fatty acid tails, so you'd have 3 fatty acids and then just be left with this glycerol molecule right here. So in the stomach, the enzyme that's responsible for that is called gastric lipase. And then if you've had any GI curriculum, you'll know that in the duodenum, bile salts are released from the biliary tree, and the point of those is going to be to emulsify the triglycerides. So remember, you've got these big hydrophobic molecules, and the tendency of hydrophobic molecules is to all sort of clump together and minimize their surface area. The bile salts are going to kind of help get into that big clump and help break everything up and spread it out, increase the surface area so that enzymes and things can all get into that space. So it'll help disperse things a little more. And those bile salts in the duodenum are also going to activate pancreatic lipase, which is another lipase enzyme that's going to help further break down these triglycerides. All right, so now we've effectively broken these triglycerides down into their pieces, right? Just like that IKEA bookshelf. We've taken it apart and now we're just sort of left with these slats that we can move across membranes. Next thing to do is take all of our pieces and put them into a moving box. And here, the moving box is going to be called a mixed micelle. And these mixed micelles are just packages of all of these lipid materials that we're trying to absorb. So we had our fatty acids and our monoglycerides that we talked about from the breakdown of those triglycerides. We also had those bile salts that were helping to emulsify everything, as well as our fat-soluble vitamins. So those are vitamins A, D, E, and K. We consumed some of those too when we took in that fatty meal. And all of those things are going to be packaged into this box box called the mixed micelle. And now for this lesson, we're really focused on following the path of those fatty acids, but just so you understand what happens to the rest of those components of the mixed micelle, the bile salts are going to be recycled in the ileum, and they'll actually go back to the liver for resecretion. So your body will reuse those bile salts over and over again. And then those fat-soluble vitamins are going to get absorbed. But what's happening to those fatty components that we're really interested in? That's going to depend on how long that fatty acid chain was. So remember, the longer that chain, the more hydrophobic it is, sort of the bigger that piece is, and the harder it is going to be to transport it and move it through all those membranes. So our short and our medium chain fatty acids are easy. Those are just going to get absorbed right into the capillaries through the jejunum. That's not really a problem. Those long chain fatty acids, though, have to take a couple of extra steps just because they're so big and clunky. So remember, those bigger pieces are still packaged up into those mixed micelles, the long chain fatty acids and the monoglycerides, and they are going to be taken up into the intestinal epithelium. Then we can empty the mixed micelle of those pieces, right? Taking the pieces of our bookcase out of our moving box, and then we can reassemble our bookcase, right? Or re-esterify our triglyceride. So we can put those fatty acids and those monoglycerides back together as a triglyceride now that we've sort of gotten through the door. So let's go over to our image and take a little bit of inventory of the steps we've covered so far. So in the intestinal lumen, which is where we are out here in the yellow, we had just taken in a big fatty meal rich in triglycerides, and then lipases cleaved up those triglycerides into their components, right? Into fatty acids and monoglycerides. Then we took those pieces, put them into our moving box, that mixed micelle, and remember we had the help of those bile salts to emulsify everything. And like we kind of alluded to, and we'll come back to in a second, cholesterol also gets packed into these micelles. Then you can see the mixed micelles with those long chain fatty acids and those bile acids and everything come into the enterocyte, and then at that point we can unpack those micelles. So those fatty acids will come out, right? And then the cholesterol will come out as well, which we'll talk about in a minute. And then now we've got all our pieces across the door, we can reassemble those triglycerides from their component parts. So we'll take those fatty acids and we'll take some glycerol and we'll put them back together and we'll reform our triglycerides now inside the enterocyte. So now that we're in the enterocyte, we need to find a way to get all of these things from the enterocyte into the blood. And that's going to involve some more packaging things up. So just like how our last moving box to get from the lumen of the gut into the enterocyte was called the mixed micelle, our next moving box to get from the enterocyte into the blood is going to be called a chylomicron. And what's going into this moving box? It's going to be triglycerides, which we just finished reassembling, as well as cholesterol esters, which I'll do in our cholesterol colors here, as well as something called apoprotein B48. Now we're going to learn a lot more about all of these different apolipoproteins, right? But the first one you're going to be introduced to is this one right here, this ApoB48. And the best way to describe these apolipoproteins, if we're sticking with our moving box analogy, is I kind of think of them like the packaging tape that's keeping that whole box together. So they're there on the outside, they're kind of these surface markers that wrap around and sort of help keep everything snug. The one that's doing that for the chylomicron and sort of keeping everything in place in the chylomicron is that ApoB48. So these chylomicrons are only going to be present in the blood in the fed state, right after you've taken in a fatty meal. And there's a molecule called the microsomal triglyceride transfer protein, called the MTP for short, or sometimes you might see it as the MTTP. It's the same thing. And the purpose of this protein is to add the ApoB48 to that chylomicron, kind of wrap everything up with that ApoB48, make sure it's packaged into the chylomicron and ready for export into the blood. Now that those chylomicrons are packaged up and ready to go, thanks to the help of the MTP, we can send them out into the extracellular space where they're going to be taken up into the lymphatics. And the pathway it's going to follow is it's going to be taken into lacteals. And remember, the flow of lymphatics, that lymphatic thoracic duct is eventually going to dump into the subclavian vein. And that's how you get from the lymphatics right back into the main bloodstream. So now we've got these chylomicrons packaged up with triglycerides and cholesterol esters floating around in the bloodstream. And there in the bloodstream, they're going to gain two more apolipoprotein surface proteins. Those are going to be ApoC2 and ApoE. For now, just understand that they sort of pick up two more surface markers once they're in the blood. So let's go back to our image here. Remember, we had sort of left off, we had re-esterified those triglycerides in the enterocyte. Now we're going to package them into a chylomicron, and that's happening in the Golgi of these enterocytes. So we'll put in the triglycerides, and we'll put in the cholesterol, which we're coming back to in a second, and we'll put on that ApoB48 kind of packing tape to keep everything snug. And remember, the molecule that's facilitating that is that MTP protein. And we load all of those things together and we form our chylomicron. Then the chylomicron will go out into the lacteal, it'll go into the thoracic duct, and eventually the subclavian vein. Now we're in the blood supply. You can see we add on 2 more of those apolipoprotein surface markers here, and we are good to go. We have our chylomicron kind of as we're gonna leave it for the next video. So let's shift gears now and talk about cholesterol absorption and how we're gonna get that cholesterol from the lumen of the gut into the blood. You understand all the hard part already. Let's just focus a little more on the cholesterol specific pathway. So we take in our big fatty meal. Not only is it full of triglycerides, but it's also full of cholesterol esters. So now we need to break down that big insoluble cholesterol ester into its pieces so that we can repackage it and move it across the membrane, just in the same analogous way to what we just did with the triglycerides. So pancreatic cholesterol esterase is the enzyme that's going to cleave those cholesterol esters into the cholesterol, as well as a fatty acid, right? So here's our cholesterol ester. Remember, this is an ester bond right here. We'll cleave it off. We'll be left with the cholesterol component and that long fatty acid tail. Then, just like with the triglycerides, we're going to take our pieces, load them into the moving box called the mixed micelle. So the cholesterol and the fatty acids and the bile salts are all going to be loaded into the mixed micelles as above, as we already talked about. And then those are floating along in the lumen. And the cholesterol specifically is going to be absorbed into the enterocyte via this transporter called the NPC1L1 transporter. And then once that cholesterol is in the enterocyte, it's going to be packaged into that chylomicron in the same way that those triglycerides were. And once it's in the chylomicron, it goes into the lymph and then into the blood, just as we saw above. It's the same chylomicron. Within that chylomicron, within the core, some of that cholesterol may be reesterified into the bigger cholesterol esters that are protected within that chylomicron core. So let's summarize it on our image here, right? We just finished taking in that fatty meal full of cholesterol, so we've got those cholesterol esters floating around in the lumen of our gut. The cholesterol esterase enzyme is going to break those apart into its pieces, cholesterol and fatty acids. We already talked about what happens to the fatty acids. Those get packaged into the mixed micelles, as does the cholesterol, and then that floats around in the lumen of the bowel for a little bit. That cholesterol specifically gets taken up via this transporter here, NPC1L1, into the enterocyte, and that name will be important later when you learn about some pharmacology of hyperlipidemia, right? This transporter is a target for some drugs. So we got our cholesterol into the enterocyte now. It's going to go to the Golgi and it's going to be packaged into the chylomicrons just like the triglycerides were. Then the chylomicrons will go out into the blood where they pick up those surface markers, all of which is the same as the pathway we just finished talking about. We just explained how we got from the cholesterol ester to the cholesterol and how that cholesterol got packaged into the chylomicron alongside the triglycerides to float around in the blood. So let's talk about a clinical implication here, which is a disorder called A-beta lipoproteinemia. And as the name might imply to you, this is going to be a defect in the beta lipoproteins. So those beta lipoproteins are ApoB48, as well as another apolipoprotein that we haven't quite talked about yet called ApoB100. It's kind of like the cousin to ApoB48, and you'll learn about it in the next video. But remember, we learned that ApoB48 is kind of like the packing tape that holds the chylomicrons all together. The reason for this defect is a defect in the microsomal triglyceride transfer protein, the MTP. That's caused by a mutation in a gene called the MTTP gene. So remember, the MTP here was in charge of loading that ApoB48 onto the chylomicron, and it kind of helped package that chylomicron together. In A-beta lipoproteinemia, you don't have a functioning MTP, which means you don't have functioning ApoB48 and you don't have a fully packaged together chylomicron, because that ApoB48 surface molecule, which is kind of under all my pen marks here, is not going to be there. So you'll end up with a decrease in chylomicrons. You're also going to to end up with a decrease in what are called VLDL and LDL. Those are very low density lipoproteins and low density lipoproteins. We haven't talked about that yet in this video. We're going to talk about them in the next video, so you can come back to this point later if you want. But the point is, just like how ApoB48 packaged together the chylomicron, that ApoB100 surface molecule is the equivalent that sort of packages together these VLDLs and LDLs. And those are packages that control sort of the flux of lipids from the liver to the peripheral cell. So without chylomicrons, we can't get lipids from the enterocyte into the blood. Without these VLDLs and LDLs, which again, I know you haven't learned about yet, but just understand, without those things, you're not going to be able to get lipids out from the liver. What that means is you're going to get a buildup in the enterocytes and in the hepatocytes due to the decreased export. We don't have the moving boxes to get all those things out of those places. As you might imagine, this is going to lead to fat malabsorption, right? We can't even take it in from the intestine. We can't get it out from the liver to the peripheral cells. It's a mess. Fat malabsorption leads to a fat-soluble vitamin deficiency, as well as steatorrhea, right, which is this fatty, greasy stools. You're also going to see a decrease in plasma cholesterol because we're having trouble absorbing. And this is going to lead to some interesting path findings. The first are these acanthocytes, and acanthocytes are spiculated red blood cells. So you can check out over here what it looks like. These are all red blood cells, and they kind of all have this like paint splatter look to them. That's because if you'll remember, cholesterol and lipids, right, are a really important part of cell membranes. You'll also see lipid-laden enterocytes, right? For the same reason we just discussed, we don't have any chylomicrons to take those lipids out of the enterocytes. So let's sum things up. We've got these big, hydrophobic, clunky fat molecules when we eat a big fatty meal. We need to break them down into their pieces, put them into moving boxes, and get them across the membranes where we can put everything back together and get them sent off to the blood. So lipase enzymes broke our triglycerides down into fatty acids and monoglycerides, and cholesterol esterase broke our big clunky cholesterol esters into, again, those fatty acids as well as cholesterol. All of those things were emulsified with bile salts, packaged into these mixed micelles, and brought into enterocytes. There we were able to reassemble those fatty acids back into triglycerides. We were able to reassemble a little bit of cholesterol too into cholesterol esters, and we packaged all of those things into chylomicrons, which was the next big package to get these lipids out from the enterocytes through the lymphatics and eventually into the bloodstream. And now that we have digested and absorbed these lipids and they're sitting in the bloodstream, in the next video we'll talk about where do they go from there. That's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our introductory lesson to lipid transport proteins and enzymes. So in the last video, we focused on how we get lipids from the lumen of our gut after we take in a big fatty meal into the blood. And we talked about how these lipids in the forms of triglyceride and cholesterol esters were loaded into chylomicrons and put into the blood. Remember, we described those chylomicrons kind of like moving boxes full of the triglycerides and the cholesterol with this sort of moving tape around it, keeping everything packaged together. That tape being apolipoproteins. And we absorbed all of those things and we put it from the intestine into the bloodstream, and that's kind of where we left it. Now in this video, we're going to talk about what happens from there. And we're going to empty out those chylomicrons. Specifically, they'll get emptied by the adipose tissue, right, fat in your body, and by the liver. And then once we've emptied out those chylomicrons, guess what? We're going to repackage things again into a different set of boxes, right? Different lipoproteins. And those new moving boxes, those new lipoproteins are going to be exported from the liver out to the periphery, to the cells that are requiring these lipids. There will also be exchange happening from these peripheral cells where they can send their lipids actually back to the liver. So take a look at this diagram. This is a lot to unpack, and we're going to spend the next couple videos talking about it. All right, but the good news is we've already covered all of this in here. This is how we took the lipids from the lumen of the intestine into the enterocyte, brought it into the lymph, and then to the blood. And we left off with our chylomicron right here floating around into the blood. And as you can see, there's about to be a lot more moving parts, a lot more lipoproteins, a lot more enzymes that are going to control the next transport action items. For this video, though, first, it's really important just to understand all of the terminology that we're going to be working with. So we're going to spend this video defining the terms, and then in the next video, once you sort of understand the foundation of, okay, what are all of these moving parts, we'll put it all together chronologically. So there are three sort of categories of definitions we're going to go through. We're going to go through lipoproteins, we're going to go through enzymes, and we're going to go through apolipoproteins. Lipoproteins, remember, were kind of those moving boxes we were talking about. They're basically just packages of triglycerides and cholesterol and cholesterol esters, as well as some other proteins and phospholipids. And you already understand the basic concept of lipoproteins because you've already been introduced to the first one we'll talk about, which are chylomicrons. Remember, these were packaged in the Golgi of the enterocytes, right, in the intestine. We took the triglycerides and the cholesterol from the diet and packed them up in the intestine and sent them to the blood. So as you're going to see, most of the triglycerides from that chylomicron floating around in the blood are going to get dumped into adipose tissue for storage, and some of those triglycerides will make their way back to the liver. And so after we unpack all of the triglycerides out of the chylomicron, it's going to be called a chylomicron remnant. And that chylomicron remnant has been depleted of triglycerides, and it's just full of cholesterol. And so that chylomicron remnant full of cholesterol is going to find its way back to the liver. All right, so chylomicrons and chylomicron remnants I sort of think of on their own island. The next four I like to think of all in conjunction. And all of these are lipoproteins that are described by their density. So you have a very low density lipoprotein, an intermediate density lipoprotein, a low density lipoprotein, and a high density lipoprotein. So let's take a look at our image. Here we have our VLDL, our IDL, our LDL, and our HDL. And as you can see, these are all kind of cousins, but they're definitely not quadruplets, okay? There are important differences between each of them. For one, as you can see, they all have different surface markers, right? Different apolipoproteins holding them all together. And we'll talk about those in a minute when we go through all the different apolipoproteins. And like we said, these are all going to be defined by their density. Now, the official way that biochemists are going to define this density is the protein to lipid ratio, right? So protein is very dense compared to big fatty lipids, which are lower density. So the more lipids in one of these lipoproteins, the lower density it is going to be. Now, that being said, when I was learning this, I didn't find that definition to be all that helpful in terms of telling me the actual ratios of triglycerides to cholesterol within each of these different particles. So I'll give you my memory device for this, and it's not a super legitimate way of thinking about it, but it does hold true. So for me, I didn't find it very helpful to think about the proteins aspect of it at all, because practically what I'm worried about is the ratio of triglycerides to cholesterol within each of these packages. As you'll see when we start to tie it all together, that's kind of the critical piece of information to take out of this. Instead, I always remembered the densities as the ratio of cholesterol to triglycerides. So lipoproteins that are higher in cholesterol are going to be higher density. Lipoproteins that are very high in triglycerides are going to be very low density. And that's kind of the way you can think about the contents of these different lipoproteins, because otherwise they're very hard to keep straight. So by the memory device that we just established, the very low density lipoprotein is mostly going to be triglycerides. So you can see that depicted here. It is our largest of these lipoproteins, and you can see it for sure has cholesterol in it, but it has way more triglycerides all throughout here. Now, if we take out some of those triglycerides, our next particle is called the IDL, or the intermediate density lipoprotein. And it's intermediate because it's about equal in proportions of cholesterol and triglycerides. If we take out even more triglycerides, we end up with the low density lipoprotein, which is mostly cholesterol, right? And now the terminology of low versus very low is something to actually remember, because as backwards and as frustrating as this is going to be to you, the low density lipoprotein is still higher density than the very low density lipoprotein, and then the intermediate density lipoprotein. All right, but as we go from VLDL to IDL to LDL, we're taking triglycerides out and we are increasing the ratio of cholesterol to triglycerides. All right, and then finally we move to our littlest lipoprotein, the high-density lipoprotein. And now this is so high density because it is mostly proteins and phospholipids, right? It's very small and packed. There's not much of anything in there. But in terms of the actual contents, once again, it has more cholesterol than triglycerides. All right, so let's go back to our outline. The first lipoprotein we just finished talking about was the very low density lipoprotein that was packed full of triglycerides from the liver. And the point of this is it's going to take those triglycerides to the periphery, so the adipose tissue and the muscle tissue. Then we talked about our intermediate density lipoprotein, or our IDL, which remember was a VLDL that was partially depleted of triglycerides, making it slightly higher density. Right now, since we've taken some of those triglycerides out, the cholesterol makes up a bigger proportion of that lipoprotein. So the point of that IDL, which has had some of its triglycerides removed already, is to continue to dump triglycerides, and it is going to dump those triglycerides back to the liver. When the IDL dumps even more of its triglycerides to the liver, right, now there are even fewer triglycerides and a higher ratio of cholesterol to triglycerides within the LDL, it's going to deliver the cholesterol to the periphery, right? So we've sort of sucked out a lot of the triglycerides. Now we've got a fair amount of cholesterol. Now that LDL is going to dump the cholesterol. And when you hear people talk about good cholesterol and bad cholesterol in your blood, LDL is considered the bad cholesterol because it is in charge of taking dietary cholesterol and dumping it out into your tissues. Compare that to the high density lipoprotein, which was our little lipoprotein that didn't have much of anything in it, but it did have more cholesterol than triglycerides in it. And the job of the high density lipoprotein is to kind of undo all the work that the LDL did. So it's going to take the cholesterol from the periphery and bring it back to the liver and lower your serum levels of cholesterol. So that's considered the good cholesterol, that high density lipoprotein. All right, so those were all of our lipoproteins, right, which were our moving boxes or our packages of lipids in different proportions. Let's go ahead and name the enzymes that are involved in lipid transport. The first enzyme we talked about in the last video, it's in the small intestine, and it's called pancreatic lipase. And remember, the purpose of pancreatic lipase was to break down those dietary triglycerides in the small intestine into their component parts, right? The monoglycerides and the fatty acids. The next enzyme in the process we'll talk about is called lipoprotein lipase. And I'm just telling you right off the bat, there's a lot of different lipases to keep track of. But this lipoprotein lipase is going to be in the blood. And what lipoprotein lipase, or LPL, is going to do is it is going to be what is responsible for cleaving the triglycerides out of the chylomicrons, right, which turn them into chylomicron remnants, and out of the VLDL, which turned it into IDL. All right, so we're not getting into all of the intricacies of this diagram yet, but just to kind of give you a sneak preview, right, here was the chylomicron out in the blood, and then here you can see lipoprotein lipase draining out all of those lipids from the chylomicron, leaving that chylomicron as a chylomicron remnant to bring cholesterol back into the liver. You can also see here that very low density lipoprotein, right, very full of triglycerides coming from the liver and going out into the blood. And here you can see lipoprotein lipase draining that VLDL of further triglycerides and fatty acids, leaving it as an IDL, partially depleted in triglycerides. And as you can see, that lipoprotein lipase is going to be upregulated by something called PPAR alpha. And that PPAR alpha will come back into discussion when we talk about the pharmacology of hyperlipidemia. So put a pin in that for now. The next enzyme we'll talk about is called hormone-sensitive lipase. And now, unlike lipoprotein lipase, which was in the blood, hormone-sensitive lipase is in the adipocytes. And in this chapter, we're going to save what happens in the adipocytes sort of for the end of our lipid discussion. So it's going to be something that we sort of dance around a little bit before we get to. But what it's going to do is it's going to take the triglycerides that have been dumped off into the adipocytes for storage, right, as you can see here coming in from the VLDL. Hormone-sensitive lipase is going to cleave those storage triglycerides into fatty acids that can be mobilized into the blood during the starved state. So it'll break apart storage triglycerides into its components, the fatty acids and the glycerol. The next enzyme to talk about is hepatic lipase, which, as the name suggests, is in the liver. So I kind of think of lipoprotein lipase and hepatic lipase kind of like buddies that each do half of a job. So we had our chylomicrons and our VLDLs that were rich in triglycerides, and lipoprotein lipase's job in the blood was to start to pull those triglycerides out. When it did that, right, it turned the chylomicron into the chylomicron remnant and the VLDL into the IDL. Now hepatic lipase is going to come and finish that job in the liver. It's going to cleave the rest of those triglycerides out of the IDLs and the chylomicron remnants. And you can see that happening in our diagram here, right? We already talked about the lipoprotein lipase starting to do that job, right, and forming the chylomicron remnants out of the chylomicrons and forming the IDL out of the VLDL. The job of the hepatic lipase then is to finish the job and empty further triglycerides out from those respective lipoproteins. Next enzyme we'll talk about is called PCSK9, and you're not going to need to know what that stands for. That's out in the peripheral cells. And the point of PCSK9 is going to be to degrade the LDL receptor. The LDL receptor controls the uptake of LDL out of the bloodstream. So PCSK9, by degrading that LDL receptor, is increasing that serum concentration of LDL. I'll show it to you on the diagram so you can keep tabs on it for later, but right here is PCSK9, which you can see degrading the LDL receptor that's controlling the uptake of LDL out of the blood. I just want to check in here and remind you that for now, all you're focused on are really getting these definitions down. And we're going to talk about the interplay of all of these arrows and everything in the next video, okay? Next enzyme we're going to talk about is called lecithin cholesterol acyltransferase, or LCAT for short. And LCAT is located in the peripheral cell. And remember, we talked about how HDL, the point of HDL is to harvest cholesterol from these peripheral cells and bring it back to the liver. So this LCAT enzyme here is going to be in the periphery, and it's going to load those cholesterol esters onto the HDL for return to the liver. So it's in charge of packing up the HDL with that peripheral cholesterol. And then the last enzyme we're going to talk about is called cholesterol ester transfer protein, or CETP. This is going to be in the blood, and the point of CETP is to trade the cholesterol esters that are in the HDL with the triglycerides that are in VLDL, as well as some that are in chylomicrons. And you can see those trades happening here on our diagram, right? Swapping the triglycerides for the cholesterol esters between the HDL and the VLDL and the chylomicrons. And there are important clinical implications for that we'll talk about in the next video. All right, so we've talked about the lipoprotein packages. We've talked about the active enzymes. Now we're going to talk about the apolipoprotein kind of surface markers that hold those lipoproteins together. And besides just holding everything together, those apolipoprotein surface markers also each have a specific function. We'll start with an example you're already familiar with, which was that ApoB48 that we first encountered in the last video. Remember, this controlled chylomicron secretion from enterocytes. And so this was present on chylomicrons, and it was also present on those empty chylomicrons, which were called the chylomicron remnants, right? It just sort of stuck around after all that emptying happened. And then remember, we said once that chylomicron got into the blood, it picked up two more apolipoproteins that we hadn't quite talked about yet. Those were ApoE and ApoC2. The point of ApoE is to control the uptake of lipoproteins into the liver. And as such, a lot of the lipoproteins that make their way back to the liver are going to have this ApoE on them. So chylomicrons, chylomicron remnants, VLDLs, IDLs, and HDLs all have ApoE on them to control their uptake to the liver. The next apolipoprotein is ApoC2, which is going to be a cofactor for lipoprotein lipase, that enzyme we already discussed. And the lipoproteins that are going to take advantage of that are going to be chylomicrons, VLDLs, IDLs, and HDLs. The next apolipoprotein is called ApoB100, and this is kind of like a cousin to ApoB48. It's another beta apolipoprotein, but as you can tell by the number, right, 100 versus 48, the ApoB100 is going to be a little bit bigger, and it's going to have an extra domain in it that controls binding to the LDL receptor. And so the lipoproteins that are going to take advantage of that are VLDL, IDL, and LDL. And then the last apolipoprotein we'll talk about is ApoA1, which is going to activate that LCAT enzyme that we talked about too, and so that's going to be on HDL. All right, so let's wrap things up. The point of this video was definitions, all right? We defined a lot of different things under three major categories. We defined a lot of the lipoproteins, which were packages for lipid transport. We talked about some of the enzymes that facilitate the process, and we defined some of the apolipoprotein surface markers. And we started to plot all of these things on our large lipid transport diagram. In the next video, we're going to pick up where we left off with chylomicrons floating around in the blood, and we're actually going to see how all of these different moving parts all fit together to control lipid transport. So spend some time with these definitions before moving on to the next video. Till then, that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on lipid transport. So our goal for this video is to hack away at this big lipid transport diagram. And now remember, we've already started to unpack this a little bit. When we talked about lipid digestion and absorption, we covered this part of the diagram right here, which I'm outlining in gray. We talked about how the lipids that we take in through our diet are packaged up eventually in chylomicrons in the intestines and are released into the bloodstream. And that was kind of where we left off. And then I just finished giving you an overview video full of a ton of definitions of all of the key players of lipid transport. And now we're going to see how all of those players fit together and how we actually get those lipids from the bloodstream to the tissues where they need to go. So remember, we had our chylomicrons, which were packed full of triglycerides and cholesterol and cholesterol esters, and those surface apolipoproteins that were holding everything together. Those had all been released into the blood, and that was where we left them. Now in this video, we're going to pick up with those chylomicrons in the blood, and we're going to cover how they get emptied by the adipose tissue and the liver, and then how those triglycerides and the cholesterol and cholesterol esters all get repackaged in the liver into lipoproteins and sent out into the peripheral tissues where those lipids are needed throughout the body. And then we'll also talk a little bit about the return pathway where lipids from the peripheral tissues can get brought back to the liver. So we're going to go through that whole big diagram on the other slide, but just so you understand the outline, we're going to talk first about just triglycerides and how those get to the periphery and the liver. Then we'll switch gears and talk about how cholesterol gets to the liver. Then once we've talked about those two things separately and how they make it to the liver where they'll get repackaged, we'll start to think about them as a unit getting exported from the liver together, and then we'll finish off by talking about that return pathway, that peripheral cholesterol activity. And it might help you to have your list of definitions from our introductory video on this topic out in front of you so you can keep track of all the different enzymes and lipoproteins and apolipoproteins that we've already been introduced to. So we followed our chylomicron full of triglycerides and cholesterol and cholesterol esters from the enterocyte right here through the lymphatics and into the bloodstream. And this right here is our capillary. The other sections of this diagram are going to be the adipocyte sort of down here, and then the liver is up here at the top of the image, just so that you can get oriented. So we'll zoom into our capillary here, and here is our chylomicron that just came from the intestine. The chylomicron is going to encounter an enzyme called lipoprotein lipase right here. And that LPL is attached to the wall of the capillary right here. And the way that it got to that capillary is it was actually synthesized in the adipocyte. And then when you take in a big fatty meal, remember, insulin is the hormone that is released, indicating the fed state, that there's lots of lipids to be dealt with. That insulin is going to encourage the adipocyte to express lipoprotein lipase, and that lipoprotein lipase will go and insert itself on the adipocyte capillary endothelium. Now remember, when we gave the definitions of all of our different apolipoproteins on the surface of all of these lipoprotein packages, one of the ones we talked about was ApoC2. And the point of ApoC2 was to act as a cofactor for lipoprotein lipase. So that chylomicron with its ApoC2 apolipoprotein right there on the surface is going to be acted upon by lipoprotein lipase. And that LPL is going to take those triglycerides that are within those chylomicrons and break it down into fatty acids and glycerol, right? Those components of triglycerides. So it's going to cleave apart those triglycerides that were inside that chylomicron and effectively empty out that chylomicron. So now we have a chylomicron that has been depleted of triglycerides, and that is called a chylomicron remnant. That chylomicron remnant has the ApoE apolipoprotein on its surface. And that's going to enable that chylomicron remnant to bind to the ApoE receptor on the surface of the liver and be endocytosed into the liver. All right, so we had our chylomicron, we had it acted upon by lipoprotein lipase, which took out some of those triglycerides, and then we were left with a triglyceride depleted, for the most part, chylomicron remnant. That chylomicron remnant goes to the liver. And then what happened to those triglycerides that were cleaved and taken out of the chylomicron? Right, they were broken into glycerol and fatty acids. That glycerol is not shown on this diagram here, but that's going to go into the liver to participate in gluconeogenesis. And those fatty acids can go one of two places. They can either go to the liver, or they can go into the adipocyte, which is primarily where they do go, to be re-esterified into triglycerides and put into storage. We're going to have a whole other video where we talk about what happens in this mysterious adipocyte down here, but for now you can just understand that those free fatty acids that are liberated in the bloodstream can be taken up into the adipocyte and then re-esterified into triglycerides for storage. All right, so now we have our chylomicron remnant, and it is in the liver, and it's been partially depleted of triglycerides by that lipoprotein lipase. We're going to finish depleting that chylomicron remnant of triglycerides with this enzyme here, hepatic lipase, in the liver. And those triglycerides and the remaining cholesterol will be dumped out of that chylomicron remnant by that hepatic lipase. All right, so that's how we get our triglycerides from the chylomicrons into the liver and into some of the peripheral cells like the adipocytes. Let's go back to our outline and inventory some of those steps. We had the adipocytes that were synthesizing that LPL, which attached itself onto the capillary endothelium of the adipocyte capillary. And the expression of that LPL on the adipocyte capillary endothelium was increased by insulin, which was a hormone that was signaling the intake of a big fatty meal in the body. Once that LPL was in place on the capillary endothelium, it was free to bind that ApoC2 cofactor, which was on some lipoproteins like chylomicrons. And the action of that LPL on the chylomicrons was to empty those chylomicrons of their triglycerides by breaking them apart into glycerol and fatty acids. And that glycerol went back to the liver, and those fatty acids were mostly taken up by adipose and put into storage as storage fat, and some of it was taken up into the liver. Then those chylomicron remnants, which were partially depleted of triglycerides, used their ApoE surface apolipoprotein to bind to the ApoE receptor on the liver. Now technically that has a name called the LRP1 receptor, and that allowed that chylomicron remnant to be taken up and endocytosed into the liver, at which point that enzyme hepatic lipase could further facilitate the emptying of those chylomicron remnants. So that's how our triglycerides get into the liver, as well as those adipocytes. Now let's talk about how cholesterol gets into the liver, part of which we just saw, right? Those chylomicron remnants still contained cholesterol and cholesterol esters. So when those chylomicron remnants were endocytosed into the liver, that brought cholesterol into the liver as well. However, cholesterol in the liver is also synthesized de novo. So back to our diagram, right? Here was the chylomicron remnant that had made it into the liver, being emptied by hepatic lipase to release its cholesterol into the liver. But then as you can see over here, another source of cholesterol in the liver is this de novo cholesterol formation. We're actually going to start with some products that we've seen plenty of times before, your old friends from the TCA cycle. We'll start with citrate. However, through a new enzyme that we haven't encountered yet called ATP citrate lyase, we're going to turn that citrate actually back into acetyl CoA. And not to throw too many crazy diagrams at you, right? But back to our old friend, the metabolism map. Here's the TCA cycle. Here's acetyl CoA. We just use that ATP citrate lyase to run this sort of weird reverse step that's not even shown on this map here to turn citrate back into acetyl CoA. And then remember, acetyl CoA is a branching off point for so many different pathways, one of which, as you'll see, is cholesterol synthesis. All right, so we formed our acetyl acetyl CoA. Now let's follow that pathway of cholesterol synthesis. There's a lot of steps you could focus on here. I'm just going to pitch the big ones to you. The first one being that acetyl CoA turns into something called HMG CoA. It stands for hydroxymethylgluteryl CoA. I've almost never seen it written out by its birth name like that. And then the big high yield step to really lock down is this rate limiting enzyme right here, HMG CoA reductase, which is going to turn that HMG CoA into something called mevalonate. This step is particularly important. It is the rate limiting step, and it is going to be downregulated not only by the presence of cholesterol, right, signaling that we don't need to create any more cholesterol de novo if we already have plenty here in the liver. It'll also be targeted by a very popular hyperlipidemia pharmacological agent called statins. We'll talk about that more in a future video, but this is the enzyme you really want to remember. Once we've made that mevalonate, there's a couple more steps in there, but eventually you'll end up down the line with new cholesterol being created de novo from those TCA cycle intermediates. So that's how you end up with cholesterol in the liver. So once we had formed that acetyl CoA from the citrate via the ATP citrate lyase, we could turn that acetyl CoA into HMG CoA. Then that important rate limiting step that was high yield, our HMG CoA turned into mevalonate via that enzyme HMG CoA reductase. And the activity of that rate limiting enzyme was decreased by both statin drugs and cholesterol. And then once we had that mevalonate, there were some more steps in there that we weren't too concerned with, where we ended up with new de novo cholesterol being synthesized. So now we have our triglycerides in the liver, we have our cholesterol in the liver. What happens once those things are in the liver? So now we have our triglycerides in the liver, we have our cholesterol in the liver. What happens once those things are in the liver? Well, they're going to get repackaged up and they're going to be exported out from the liver and sent to the periphery. So we've been using this sort of long analogy of these lipoproteins being packages carrying lipids. Sticking with that analogy, you can think of the liver as sort of the central processing hub, right? Everything has to come into the central processing hub. We'll repackage everything up and then we'll send it out where it needs to go. For our next step, we're going to revisit our old friend, the microsomal triglyceride transfer protein, or the MTP. You'll remember we first saw this when we were talking about the absorption of lipids, right? If you check out down here, remember the MTP was what loaded that ApoB48 onto the chylomicron, and in that way facilitated the packaging of the chylomicron for export. So it's really good at facilitating packages for export. That's sort of the job of the MTP. And it's going to be doing that again here. This time the package that it's going to be assembling is the VLDL, the very low density lipoprotein. And in our definitions video, we talked about how this is very low density because it's packed full of low density triglycerides. So the MTP is going to pack a lot of triglycerides into this new VLDL package. It's also going to package in some cholesterol, and it'll load on the ApoB100 surface apolipoprotein to help bind everything together, and it has an important function down the line that we'll talk about later. For now, it's just along for the ride. Now that VLDL has been packaged up and it is ready for export into the blood. And it's in the blood where it will pick up two more surface apolipoproteins, ApoC2 and ApoE. This should all feel very analogous to the chylomicron secretion. And just like with the chylomicron, the next step is going to be lipoprotein lipase, right? That same lipoprotein lipase that was in charge of emptying triglycerides out from chylomicrons. Now it's going to work on the VLDLs. It'll bind that ApoC2 cofactor, and it'll empty out those triglycerides by cleaving them apart and turning them into fatty acids. And remember, those fatty acids go into the adipocytes for storage. Just like how the chylomicron that had been depleted of triglycerides got a new name called the chylomicron remnant, now our VLDL that's been depleted of triglycerides is going to get a new name, and that is called an IDL, or an intermediate density lipoprotein. By taking out some of those triglycerides, we've increased the density of this lipoprotein. And again, thinking back to the path of that chylomicron, right? Once we had the chylomicron remnant that had been partially emptied of triglycerides, hepatic lipase sort of finished the job and continued to empty out more triglycerides. Again, the same thing's going to happen here, right? So we have our IDL, which was the VLDL that had been partially depleted of triglycerides. Now hepatic lipase is going to come in and finish the job, and suck out even more triglycerides, leaving us with an LDL, a low-density lipoprotein. So as those triglycerides got pulled out, we went from very low density to intermediate density to low density. And so since these LDLs have had a lot of their triglycerides removed, they're very densely packed with cholesterol. And that's something we discussed in our definitions video. So we've got these LDLs packed with cholesterol, ready to deliver them to tissues that need cholesterol. Now, important to just realize here, remember, hepatic lipase is hepatic. It's a liver enzyme. So that IDL actually gets taken up by the liver via the ApoE receptor, same ApoE receptor that was dealing with the chylomicron remnant, bring that IDL into the liver where hepatic lipase can do its job. And these steps up here, you can just see in a little more detail fleshed out how that IDL gets depleted of its triglycerides by that hepatic lipase, right? The ApoE receptor, the same one that was involved with the chylomicron remnant, is going to be involved here, facilitating sort of the docking of the IDL at the liver for that hepatic lipase to work and emptying some of those triglycerides back into the liver. All right, so we partially depleted our VLDLs of triglycerides, turning them into IDLs. We continue to deplete of triglycerides via hepatic lipase, turning it into LDL. And now we have this LDL, low-density lipoprotein that is packed up with cholesterol, ready to deliver that cholesterol to the peripheral tissues where it might be needed. That ApoB100 surface apolipoprotein is going to facilitate the docking of LDL onto the LDL receptor. And this LDL receptor is going to be in any peripheral tissue that might need those lipids. So that could be the liver, kind of is depicted up here, but it could be any peripheral cell as well. The LDL binds the ApoB100 and takes it up into the cell. And then that PCSK9 enzyme, which we talked about in our definitions video, is going to facilitate the degradation of that LDL receptor. That has important pharmacologic implications, which we'll talk about in a future video. All right, let's get back to the outline and talk about this latest chunk of activity that we've been talking about, which was the export of triglycerides and cholesterol from that liver central processing hub. We had our friend the MTP facilitating the packaging of our latest lipoprotein, the VLDL, the very low density lipoprotein. And that was made up of ApoB100, the surface apolipoprotein, triglycerides, cholesterol, and cholesterol esters. Remember, like we talked about in our definitions video, the density of the lipoproteins describes the protein to lipid ratio. But in that video, I told you sort of the best way, at least for me to think about it, was the more triglycerides there were, the lower the density. So our lowest density lipoprotein, the very low density lipoprotein, was the one that was packed the most full of triglycerides, with the understanding that as we drain the VLDL of triglycerides, we will eventually increase the density of these lipoproteins. So the MTP packages together the VLDLs and it gets secreted out into circulation. It'll pick up two more surface apolipoproteins, ApoC2 and ApoE. Then that lipoprotein lipase in the capillaries again is going to bind that ApoC2 cofactor on the VLDL and do its activity. A slight nuance to just be aware of though, here we're not only focused on capillaries of adipocytes, but also capillaries around muscle cells. The nuance here is that the expression of lipoprotein lipase in the adipocyte capillaries is going to be sensitive to insulin, whereas the LPL expression on muscle capillaries is not going to be dependent on insulin. That insulin activity is really focused on fat storage in the adipocytes. In the muscle, it just needs a constant amount of LPL activity because it's not storing any of those triglycerides as fat. And remember, the point of that LPL was to drain the triglycerides from the VLDL, turn it into IDL. Then in the liver, that hepatic lipase was going to even further deplete those IDLs of triglycerides and turn them into LDLs. And those cleaved triglycerides, right, got cleaved into their fatty acid components, which went to the adipocytes and were stored as triglycerides, which is a process we'll talk about in a future video. So then we were left with that LDL, which was really packed full of cholesterol since we had taken a lot of those triglycerides out. That LDL receptor bound that ApoB100 surface apolipoprotein on the LDL, and that facilitated the endocytosis of the LDL particle receptor complex up into peripheral cells, and that could be the liver or any peripheral cell that needs cholesterol for its membrane or what have you. All right, so we've gotten our triglycerides to the periphery, we've gotten our cholesterol to the periphery, but we can also, right, bring cholesterol out from the periphery back to the liver. And let's talk about how that happens. That's going to happen via a particle called HDL, or high density lipoprotein. This is like a scavenger protein that's going to go scavenge cholesterol from the periphery and bring it back to the liver. Now, HDL has a surface apolipoprotein on its surface called ApoA1. And the point of the ApoA1 apolipoprotein is to activate an enzyme called LCAT, or lecithin cholesterol acyltransferase. And this LCAT enzyme is going to be what packs up the HDL with all of that peripheral cholesterol. However, there's a protein in the blood called the cholesterol ester transfer protein, or CETP. And the job of CETP is to trade the cholesterol that's in the HDL, right, that's been harvested from the periphery and is living inside the HDL, with the intention of going back to the liver. It'll trade the cholesterol esters inside that HDL for triglycerides in some of the other lipoproteins in the blood, like chylomicrons and VLDL. So check it out here, right? You've got your chylomicron and you've got your VLDL. Both of them are very high in triglycerides. And then here you've got your HDL, right, which is high in cholesterol and cholesterol esters. So instead of just letting the HDL, which is trying to do its job of collecting cholesterol from the periphery and just bringing it back to the liver, CETP is going to kind of intervene and try and make this trade, where it'll dump the cholesterol esters out of the HDL and load it either into the chylomicron or the VLDL, and then it will pack the HDL full of triglycerides. So the CETP has gotten in the way of this good process of the HDL trying to bring peripheral cholesterol out of the circulation, out of the blood. Instead of letting it go back to the liver in the HDL, it's putting it back out into the circulation, removing it from the HDL and putting it into these particles, these VLDLs and chylomicrons. So in patients with hyperlipidemia, this CETP activity is a big problem, right? It's getting in the way of HDL trying to remove all that extra cholesterol from the circulation. It's snatching it out of the HDL and putting it back into the periphery by loading it into the chylomicrons and into the VLDLs. And the more cholesterol that's in the blood, the more this is a problem, because now you're going to have an HDL that is low in cholesterol and high in triglycerides due to the activity of the CETP. When that HDL that we've bloated up with triglycerides gets into the liver, it's going to be acted upon by hepatic lipase. Remember, hepatic lipase sucks triglycerides out of these lipoproteins. So if we have an HDL that's low in cholesterol and is just bloated up with triglycerides, it gets acted upon by the hepatic lipase, whose whole job is to suck the triglycerides out of it. All of a sudden you're left with an HDL with nothing left, and it just shrivels up and disintegrates. So the activity of CETP in people with hyperlipidemia means that you get these HDLs that are bloated up with triglycerides instead of the cholesterol like they're supposed to be. That cholesterol gets sent back out into the blood, which is bad in patients with hyperlipidemia. Those triglyceride bloated HDLs end up coming back to the liver, get acted upon by hepatic lipase, which effectively digests them up into nothing. And so that's why patients with hyperlipidemia, not only are they high in LDL, that bad cholesterol, but that's why they're also low in HDL, that good cholesterol, because that CETP activity in combination with that hepatic lipase activity is going to destroy those HDLs. Okay, that was exhausting. Let's wrap it up. So we had chylomicrons floating around in the blood that were full of triglycerides and were full of cholesterol and cholesterol esters. Through the activity of lipoprotein lipase on the capillary endothelium of adipocytes, we were able to remove some of the triglycerides from those chylomicrons, turning them into chylomicron remnants. Those chylomicron remnants, which were partially depleted in triglycerides, still had all their cholesterol in them, went back to the liver, where hepatic lipase was able to further empty them from triglycerides and deposit the rest of that cholesterol right there into the liver. So we've got triglycerides in the liver and some triglycerides which had also been deposited out in the periphery into adipocytes. And we got our cholesterol to the liver. And remember, we could also make cholesterol de novo in the liver from some TCA cycle intermediates. The important enzyme to remember from that was HMG CoA reductase. Once we were in the liver, that was sort of our central processing hub, where we were going to take all of these parts, repackage them into a new lipoprotein called VLDL, and that was facilitated by the MTP. We sent that VLDL package out into the blood. Again, our friend LPL was going to pull some of the triglycerides away from it, turning our VLDL into an IDL, and then hepatic lipase was going going to take even more triglycerides out, turning that into an LDL. That LDL had been very depleted in triglycerides, was very rich in cholesterol and cholesterol esters, and it was free to go deliver those cholesterol and cholesterol esters to the peripheral tissues, be that the liver or any other cell that needs cholesterol, for example, for its membrane. Then we talked about our hero, HDL, that good cholesterol, which was in charge of scavenging cholesterol from the periphery. Our enzyme LCAT was going to pack up that HDL with that cholesterol and bring it back to the liver. And then we talked about how in hyperlipidemia patients, we lose some of our HDL heroes due to the activity of the cholesterol ester transfer protein, the CETP, which is going to make a sneaky trade, trading triglycerides from VLDL and chylomicrons for the cholesterol esters that the HDL is so valiantly trying to return to the liver. And why this is no good, especially in patients with hyperlipidemia, because it depletes them of their much needed HDLs. Luckily for these hyperlipidemia patients, we have a ton of pharmacology that we can throw at this problem. And that's what we're going to talk about in the next video. Till then, that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on hyperlipidemia pharmacology. So we've done a lot of work this chapter familiarizing ourselves with the pathways of cholesterol and triglyceride as they get absorbed and digested, brought into the liver, and then delivered out to peripheral tissues. Now that we're familiar with all of that, we can start talking about the very high yield drugs that you will see pretty much every day in your clinical practice. And those are the drugs to treat hyperlipidemia. All right, we're gonna start off with our bile acid resins. And some of the representative drugs that you might see listed by name within this class are celestipol, cholestyramine, and colesevelam. And the way that these are going to work is they are going to bind to bile acids in the actual lumen of the intestine. So if we go ahead and take a look at our image, remember down here was our intestinal cell, our enterocyte. And here was the lumen of the gut, and then here's the enterocyte right here. We talked about how these bile acids here in the lumen of the intestine help to emulsify and facilitate the absorption of lipids from the lumen of the intestine in through the enterocyte into the blood and ultimately to the liver. So you can see here that the job of the bile acid resins is going to be to sequester these bile acids in the lumen of the gut so that they're not being reabsorbed and recycled for reuse. They're going to stay in the lumen and follow the whole bowel down and just get excreted. So we're excreting those bile acids and we are interfering with the absorption of lipids, which is facilitated by those bile acids. And then on top of all of that, your body needs to make more bile acids to replace the ones that aren't being recycled. So existing cholesterol is going to be used to synthesize new bile acids, and that'll take more cholesterol out of your circulation. So all of that is to say that these bile acid sequestrants are going to decrease the cholesterol levels in the liver. And the effect that that's going to have is it is going to promote more LDL receptor expression. Your liver is going to have to get its cholesterol from other places like the bloodstream. So one of the tools the liver has is to pull cholesterol out of the blood that's pre-existing by increasing the LDL receptor expression. However, the reason that these drugs aren't, the first line gold standard drugs is because your liver has more than one way of increasing its cholesterol levels. So yes, it can absorb it from the blood, but remember, it can also increase its de novo cholesterol synthesis. And that's going to limit the LDL lowering efficacy of this drug. When you increase that de novo cholesterol synthesis in the liver, you're going to promote VLDL expression. And remember, VLDLs are very high in triglycerides. So yes, these bile acid resins lower LDL, and they'll also increase your HDL slightly. But as you can see here, that LDL lowering efficacy isn't as strong as it is for some of these other drugs, and it actually increases the serum level of triglycerides. From an adverse effects standpoint, what you're thinking about here is abdominal bloating and cramping, right? GI upset symptoms, and that's due to fat and fat-soluble vitamin malabsorption. The other thing to look out for is that bile acid resins can decrease serum drug levels of things like digoxin and warfarin, right? We're interfering with the absorption of drugs and enterohepatic recycling of these drugs. So you have to be careful when you're using these because patients who are taking these drugs, their doses might become subtherapeutic. In fact, studies have even shown that you you can use bile acid sequestrants to treat toxicity from things like digoxin. All right, moving on to our next class, we have the cholesterol absorption inhibitors. And the big one here to remember by name is ezetimibe. Remember, again, we're still sort of focused on the intestinal lumen and the enterocyte. And remember, the transporter through which cholesterol is absorbed from the lumen of the gut into the enterocyte is called the NPC1L1 transporter. And ezetimibe is going to block that transporter, and as such is going to prevent the absorption of cholesterol from the lumen of the intestine. And so it'll just go off to get excreted instead. So we'll decrease that NPC1L1 cholesterol transporter activity and decrease actual cholesterol absorption right there at the source in the bowel. This is again more of a second line drug, and its efficacy is limited in a very similar way to the bile acid resins. Again, we're decreasing the overall cholesterol pool, but again, the flip side of that is then de novo cholesterol synthesis from the liver is going to kick in and sort of limit that efficacy there. And you might get a mild increase in HDL, or it might stay the same. You also might get a mild increase in triglycerides, or it might stay the same. The adverse effects here are diarrhea, again, GI upset, and you might see an elevation in liver function tests as well. All right, our next drug class is kind of the main event for this video. These are the HMG CoA reductase inhibitors, more commonly known as statins. So most of these drugs end in statin. Atorvastatin, rosuvastatin, simvastatin, it's right there in the name. So remember, with the first two drugs, their efficacy was limited by the fact that de novo cholesterol synthesis was being cranked up in response to this decreased absorption. Statins are the solution to that problem. They are going to inhibit HMG CoA reductase, which is that rate-limiting step in de novo cholesterol synthesis. And by blocking this de novo cholesterol synthesis, we're going to decrease the cholesterol content in the liver. Since de novo cholesterol synthesis is out of commission, the liver's only choice is to increase the expression of the LDL receptor and take in cholesterol from the blood. And that'll lower serum LDL. So by inhibiting that HMG CoA reductase, we're reducing the de novo cholesterol production in the liver, which gives the liver no other choice but to take that cholesterol in from the blood, since it can't make it itself, and recycle that LDL by increasing that LDL receptor expression. So we're going to decrease LDL majorly. We're also going to increase HDL, that good cholesterol, and we'll even decrease our triglycerides a little bit too. Now, the adverse effects for statins are very high yield because statins are very high yield drugs. The first adverse effect to keep in mind is hepatotoxicity, and this might manifest itself as an elevation in liver function tests. And then the major high yield side effect to know is myopathy, and this is made even worse when you combine these drugs with fibrates or niacin, which are two other drug classes we'll talk about further down the line. And this statin-induced myopathy is not something to mess around with. One thing to look for in a test question about this that might tip you off is an increase in creatine kinase. And these can get up to 10 times from normal. And so before you start statins, you want to take a baseline creatine kinase level so that you can actually monitor for this effect. And this is serious. This can even lead to rhabdomyolysis. So if patients develop myopathy or rhabdo, right, these spikes in CK, you need to discontinue them from the statin. All right, so we've got statins, which are really our first line weapon against high LDL. Now let's talk about one of our major weapons against triglycerides, which are going to be fibrates. And fibrates include drugs like gemfibrozil and fenofibrate. And the way that fibrates are going to work is they are going to upregulate this PPAR-alpha transcription factor that specifically promoted the expression of LPL lipoprotein lipase on the endothelium of the adipocyte capillary. And that lipoprotein lipase was in charge of cleaving triglycerides out of things like VLDL into fatty acids, thus decreasing the serum triglyceride concentration. So we've upregulated that PPAR-alpha transcription factor, which upregulates the expression of lipoprotein lipase and cleaves triglycerides. The other thing PPAR-alpha does, which isn't shown in that diagram, is it actually will even help promote HDL synthesis. The major effect of fibrates is going to be a large drop in triglycerides, but we'll also have increases in HDL and decreases in LDL. One of the side effects, which we've already talked about, is that myopathy, which is made worse in combination with statins. Another adverse effect you might see, however, is cholesterol gallstones. The reason here is that these fibrates are going to decrease the activity of 7-alpha hydroxylase, which is an enzyme involved in bile acid synthesis. Since we have decreased bile acids in the bile, we're going to have an increased ratio of cholesterol to bile acids, and this leads to a supersaturation of bile with cholesterol and not enough bile acids to emulsify them. That leads to cholesterol gallstones. Now the gears might be starting to turn, and another drug you might see these cholesterol gallstones with are bile acid resins. It sort of goes into that GI upset category, but for both of those, just think when you're interfering with bile acids, you can get predisposed to gallstones. So fibrates and bile acid resins are the two for that. Next drug class on our list is sort of a newer drug class in the grand scheme of things. These are the PCSK9 inhibitors. And they all have funny names, sort of with this occumab suffix, evolocumab and alirocumab. And if you remember, the function of PCSK9 normally is to promote the degradation of the LDL receptor. PCSK9 inhibitors, therefore, are going to stop PCSK9 from breaking down that LDL receptor. So by inhibiting the inhibitor of the LDL receptor, we are promoting the expression of the LDL receptor, which is going to take LDL out of the blood. So it's sort of a tricky double negative mechanism. Just remember that the upshot of all of this is to promote the expression of the LDL receptor. So by inhibiting that PCSK9, we are decreasing the degradation of the LDL receptor, which leads to an increase in LDL clearance from the blood, right? Because it's being taken up by those LDL receptors. So PCSK9 inhibitors are going to very greatly decrease the LDL, they'll increase HDL, and they'll decrease triglycerides. The adverse effects here are myalgia, as well as neurocognitive effects like delirium and dementia. Next drug class we'll talk about is niacin, that is vitamin B3. And niacin decreases the activity of hormone-sensitive lipase. Hormone-sensitive lipase is in the adipocytes, and under normal circumstances in the adipocytes in response to hormones such as glucagon, promotes the degradation of triglycerides into fatty acids, free fatty acids that can be released out into the blood. So obviously, if we are inhibiting the activity of hormone-sensitive lipase by using niacin, we are decreasing lipolysis. The other thing that niacin does is it inhibits the formation and packing of VLDL. So niacin is going to decrease lipolysis by decreasing the activity of hormone-sensitive lipase, and then it will also decrease VLDL synthesis. This will moderately decrease the LDL, moderately increase the HDL, so it's a pretty good option for increasing that HDL, and decrease serum triglycerides. One of the NBME's sort of pet favorite adverse effects to ask about with niacin, there are two that they really like, but that's prostaglandin-mediated flushing, as well as hyperuricemia. Both of those are ones I would really lock into memory. So a vignette might describe this patient who was just started on a new cholesterol drug, and they probably won't tell you what it is, and then they'll describe that this patient gets very flushed and hot after they take this new drug. They might ask you what you should do about that, and the answer is to give them NSAIDs, because this flushing is prostaglandin mediated. Hyperuricemia is the other one that they really like to test, and this might come up in a vignette about a patient who develops gout after starting a new cholesterol medication. And then finally, niacin may also cause hyperglycemia. Last drug class we'll talk about are fish oil, or more broadly omega-3s. They're not super highly tested on the NBME exam itself because there's no great explanation for the mechanism of action. They think it works by decreasing free fatty acid delivery to the liver, and that it decreases hepatic triglyceride synthesis enzymes. That being said, you'll see these clinically all the time, especially because they're available over the counter, and they do help. They do increase the LDL, increase HDL, and decrease triglycerides. And they're pretty well tolerated. The only adverse effects are nausea and poor taste. So to recap, we had a bunch of different classes of drugs The king of hyperlipidemia by far are the statins. Those are your best agent for lowering LDL. And remember, the big adverse effect to watch out for here was that statin-induced myopathy. Our big weapon against hypertriglyceridemia were those fibrates. Those also caused myopathy, which was worse when combined with statins, as well as some cholesterol gallstones. And then our best option for increasing HDL was that niacin. And remember, those had some unique side effects that they liked to test, like the prostaglandin-mediated facial flushing that was made better with NSAIDs, as well as the hyperuricemia. Then we had a couple other classes like these bile acid resins and these cholesterol absorption inhibitors that worked in the gut. We had our fancy new PCSK9 inhibitors, more expensive and not as commonly used as statins. Then we had our over-the-counter omega-3 fish oil. They were a good supplemental tool for some of these other prescription agents. And that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on familial dyslipidemias. Now we're going to talk about some inborn errors of lipid metabolism and lipid transport, which are the familial dyslipidemias. So these are autosomally inherited defects in lipid transport lipoproteins, apolipoproteins, receptors, or enzymes. And each of these familial dyslipidemias has a name, but they're also classified by number, type 1, 2, 3, 4, and then there's also a type 5, which is pretty rare. And these are called the Fredrickson classes, or it's the Fredrickson classification system. We're only going to be talking about types 1 through 4 today. Those are the highest yield for your step exam. So familial dyslipidemia type 1 is called familial hyperchylomicronemia. This is inherited in an autosomal recessive fashion. And this is going to be a defect either in LPL, which is lipoprotein lipase, or else ApoC2. Now, just to remember what those things are, remember lipoprotein lipase is what is in charge of cleaving triglycerides within chylomicrons and turning them into chylomicron remnants. The other thing it does is it cleaves the triglycerides out of VLDLs and turns them into IDLs. And then remember, ApoC2 is the cofactor for lipoprotein lipase. So back to our diagram, right? You can see in the key here, this little brown Pac-Man is lipoprotein lipase. You can see it right here. You can see it right here, and you can see it cleaving out triglycerides from the chylomicron, turning it into the chylomicron remnant, and from the VLDL, turning it into the IDL, right, and liberating those fatty acids. And when you zoom in and look at that chylomicron and that VLDL, you can see the little pink ApoC2 right there on the surface that helps to facilitate the action of the LPL. So when there's a defect in lipoprotein lipase, LPL, or else in that ApoC2, both of those are going to have a similar effect. You're going to get a buildup of chylomicrons because that LPL isn't active either by itself or because it's missing its cofactor to break down those chylomicrons into the chylomicron remnant. Because we're not cleaving out these triglycerides from these chylomicrons, you are going to see a massive increase in serum triglycerides, as well as either a normal or perhaps a slightly elevated total cholesterol. And so one of the clinical features are what are called xanthomas. And xanthomas, broadly as a category, are just deposits of lipids in the skin. And so throughout the slide here, I have a couple of different examples of types of xanthomas. And the kind that you see specifically in type 1 are eruptive xanthomas. And these are going to be yellow papules, maybe with an erythematous border around the outside. They almost look like large pimples, but they can be tender or else very itchy. And they're usually on the extensor surfaces or else the back and the buttocks. So those are eruptive xanthomas. Other things you might see are hepatosplenomegaly and recurrent pancreatitis. And this hepatosplenomegaly and recurrent pancreatitis is due to macrophage phagocytosis of lipoproteins within these organs. However, very importantly and unique to type 1 familial dyslipidemia is that there is no increased risk in atherosclerosis despite the fact that we have this increase in lipids. That's an important fact, although it is one that's a little bit difficult to test on the NBME. However, you know that the NBME loves talking about risk factors for different diseases. And if a patient comes in with hyperchylomicronemia with a heart attack, that hyperchylomicronemia is not going to be the largest risk factor for them developing that heart attack. That would be a trap answer. In most patients here, the morbidity and mortality is going to be due to this pancreatitis. Now, one thing that the NBME loves to test about type 1 familial dyslipidemia is this overnight plasma test, on which you will see a creamy supernatant. So here is our image right here. And an overnight plasma test is where they basically just take the blood and leave it in the fridge overnight and see what happens. And what happens in a patient with familial hyperchylomicronemia is you're going to see right here this creamy supernatant. Those are lipids that rise to the top of the tube. And oftentimes they will even give you this picture explicitly. And this is very specific for familial hyperchylomicronemia. And you can do overnight plasma tests for some of these other familial dyslipidemias, but they won't be very specific. In type 4, which is hypertriglyceridemia, which we'll talk about later on, right, it's pretty easy to mix up hypertriglyceridemia, which has very elevated triglycerides, again with this hyperchylomicronemia, which also has very elevated triglycerides. But the supernatant layer there is going to be turbid and not creamy. Oftentimes, if they're giving you a picture of the tube, it's probably to show you a creamy supernatant. All right, let's move on, talk about type 2 familial dyslipidemia. This is called familial hypercholesterolemia or familial combined hyperlipidemia. And that's broken into types 2A and 2B. Hypercholesterolemia being type A, combined hyperlipidemia being type B. Both of these are inherited in an autosomal dominant fashion. And the pathogenesis of this disease is an absent or defective LDL receptor or else ApoB100. And it's not like one is defective in type A and one is defective in type B. It can be either of these defective in both types. The difference between A and B is going to be what you see actually as the elevated lab values. So remember, the LDL receptor is what facilitates the uptake of LDL, which is rich in cholesterol, into the liver. And our little orange ApoB100 apolipoprotein on the surface there is what facilitates that binding to the receptor. So naturally, if either one of those things is defective, you're going to see an increase in LDL as well as an increase in cholesterol. You're going to see a mild increase in cholesterol in type 1A and a larger increase in cholesterol in type 2B, massively elevated total cholesterol. So in both 2A and 2B, you see an elevation in LDL and an elevation in cholesterol, albeit to different degrees. And then in type 2B only, you're also going to see an increase in VLDL and triglycerides. Now, in both types, you're going to see xanthomas, but different types of xanthomas. So in 2A, you're going to see tendinous xanthomas. And here are tendinous xanthomas right here. And these are going to be these firm nodules that you're seeing right here on the fingers. These are located within the tendons, specifically extensor tendons, commonly in places like the hands or the Achilles tendon. And they like to test that Achilles tendon xanthoma with type 2A. The other thing they like to test with type 2B are xanthelasmas. You can see xanthelasmas right here. They're these flat yellow plaques, usually on both sides of the inner corner of the eye. And then in both types 2A and 2B, you will be seeing early atherosclerosis, right? Compare that to type 1. So something to think about if someone comes in with ASCVD, which is atherosclerotic cardiovascular disease, at a very young age. Things like young heart attack, young stroke, young peripheral artery disease. And then the last thing they might describe is corneal arcus. This is corneal arcus right here, and these are lipid deposits in the eye. You can see this patient has brown eyes, but there's this blue-gray ring around the outside. That is the lipid deposit. Don't go thinking everyone you see in the clinic who has corneal arcus has a familial dyslipidemia. Pretty much everyone over the age of 80 or so is going to have this. It's also a part of normal aging. All right, moving on to type 3 familial dyslipidemia, we have dis-beta lipoproteinemia. This is inherited in an autosomal recessive fashion. And you can remember that the odd numbers are autosomal recessive and the even numbers are autosomal dominant, if that helps you at all. And the defect here is going to be in that ApoE apolipoprotein. If you remember our ApoE here from our introductory video to these apolipoproteins, controlled liver uptake and was on the surface of all of these different molecules, the chylomicrons, and therefore the chylomicron remnant, the VLDLs, and the IDLs, as well as the HDLs. So defective ApoE means defective liver uptake, meaning increased serum concentration of things like chylomicrons, VLDL, and increased serum concentrations of those lipoproteins means that there's an increased serum concentration of triglycerides and cholesterol. You're also going to see xanthomas with type 3, and the type of xanthoma you're going to see here, there are two of them. There's the tuberoeruptive and the palmar xanthoma. So the tuberoeruptive xanthomas are like the eruptive xanthomas, which we talked about with type 1, but they also have a tuberous component, and I can show you on the picture here. These are sort of this firm yellow. They're less erythematous around the outside as compared to something like an eruptive xanthoma, and they're also typically painless as opposed to the tender or itchy eruptive xanthoma. And tuberous xanthomas are going to be located in pressure areas like the knees or the elbows or the buttocks or maybe the heel. And so this is a picture of a tuberous xanthoma. A tuberoeruptive xanthoma is going to be something like this plus an inflammatory component. That's that eruptive component, like the eruptive xanthomas that we saw in type 1. The other thing you'll see with type 3, which is unique to type 3, are palmar xanthomas, which again are those yellow xanthomas on the palms of the hands. And again here in type 3, you're going to see atherosclerosis in these patients at an earlier age. And then type 4 familial dyslipidemia is going to be called hypertriglyceridemia. This is inherited in an autosomal dominant fashion, and this one's kind of unique from the other ones. Instead of it being a defective lipoprotein or apolipoprotein, this is the liver basically overproducing these VLDLs and pumping them out into the bloodstream. So you're going to have a massive amount of VLDLs in the bloodstream, and remember VLDLs are mostly rich in triglycerides. So your total cholesterol might be normal or it might be slightly increased, but remember when you look at the makeup of VLDLs, they are mostly triglycerides with some cholesterol. You're also going to see these tuberoeruptive xanthomas that we described for type 3, again in type 4. Another clinical manifestation is acute pancreatitis, and you mostly see that when the level of triglycerides pop up above 900 to 1,000. When they're really massively elevated, that can cause pancreatitis. And along that vein, you can also see insulin resistance, which leads to hyperglycemia. A lot of these are going to be managed in a pretty similar way. You're thinking statin therapy, and you're going to tailor that just based on their age and their LDL levels and their ASCVD risk. Remember, statins were HMG CoA reductase inhibitor drugs that inhibited de novo cholesterol synthesis in the liver, sort of our first-line agent for hyperlipidemia. And then, of course, as is the principle with high cholesterol, you can also manage with diet and exercise. All right, so let's wrap things up. There are 5 types of familial dyslipidemia, but for your step 1 exam, there are really 4 types to focus on. Type 1 is called hyperchylomicronemia due to a defect in lipoprotein lipase, or else that ApoC2 cofactor. That caused an increase in chylomicrons. Type 2 familial dyslipidemia was broken into types 2A and 2B. Those were called familial hypercholesterolemia, or else familial combined hyperlipidemia. Both of these were due to defects in either the LDL receptor or the ApoB100 apolipoprotein that helps to facilitate binding to the LDL receptor. Type 3 was dys-beta-lipoproteinemia. This was a defect in ApoE that caused a buildup in chylomicrons as well as VLDLs. And then type 4 is familial hypertriglyceridemia. This was that hepatic VLDL overproduction that caused massive increases in VLDL and triglycerides. And just to hammer this home, in type 1, the lipoprotein that was primarily elevated were chylomicrons, versus in type 4, it was primarily VLDLs. And we manage these high lipids like we manage high lipids for pretty much everyone with statins, diets, and exercise. That's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on adipocytes and lipolysis. So this is our big lipid transport diagram that we've been talking about a ton throughout this chapter. And you'll kind of notice we skipped over quite a bit what's going on down here in the adipocyte, the fat cell. We basically just said that in the fed state, these fatty acids go in and are stored as triglycerides, and then when it is time to liberate those energy stores, they can be released from the adipocyte. And that was kind of where we left it. And now we're going to get into the nitty-gritty of that a little bit more. So as you guys are familiar with, in the fed state, the hormone that is driving that fed state is insulin. When you take in a big fatty meal, insulin is released, and that is going to promote lipogenesis, or energy storage. So not only is insulin in charge of bringing glucose into cells via the GLUT4 transporter, like we talked about in earlier chapters, it also is going to mediate triglyceride storage in adipocytes. And then in the fasting state, when it's been a while since our last big fatty meal, the hormone that mediates that is glucagon, and that will trigger the cleavage of those storage triglycerides within the adipocytes and liberate free fatty acids as well as glycerol in a process called lipolysis, right? Remember, your triglyceride was just this glycerol backbone with 3 fatty acid tails. So we'll break that apart and liberate those all out for energy. So let's start by talking about lipogenesis, or the storage of triglycerides. Like we alluded to, this is happening in the fed state and it is stimulated by insulin. On the flip side, signals of a low energy state are not going to favor energy storage. They're going to favor energy release. So things like glucagon, epinephrine, norepinephrine, ANP, and BNP are all going to decrease lipogenesis. Those are going to favor lipolysis instead, which we'll talk about in the back half of the video. And the best way to think about this epinephrine, norepinephrine, ANP, BNP situation is that those are all things that are increased during exercise. And during exercise, we can't be taking energy away, sequestering it out of the blood and putting it into storage. We need to make sure we have enough energy to do our exercise. So that should hopefully at least make sense to you with the epinephrine and norepinephrine. And then remember, ANP and BNP are atrial and brain natriuretic peptides. They are released from cardiomyocytes in response to ventricular stretch. So when your heart is working hard, it's pumping a lot of blood, it's stretching out, these things will be released. And other than hypertension, as you classically think of it, the other situation where these are going to be increased is exercise, increased cardiac output. All right, so let's talk about lipogenesis on our diagram here. First, we got to talk about how these fatty acids from the bloodstream actually get into the adipocyte. So remember, chylomicrons and VLDLs are both going to be acted upon by lipoprotein lipase, which will cleave out their fatty acids. These fatty acids are going to go into the adipocyte from the blood via these fatty acid transport proteins. And not only are these fatty acid transport proteins sort of the gateway for these fatty acids to get into the adipocyte, but they're also going to facilitate the activity of this next enzyme called acyl CoA synthetase. And what this acyl CoA synthetase is going to do is it's going to conjugate those fatty acids that we just brought in to a CoA. So a fatty acid plus a CoA makes an acyl CoA. And you'll see as we move through this chapter that this theme of conjugating and activating these fatty acids by adding a CoA group is going to be a common theme. All right, now remember, we're trying to make triglycerides, and like we talked about, the components of the triglycerides are a glycerol backbone and the fatty acid tails. So we have the precursor for our fatty acid tail component, but we still need the precursor for our glycerol backbone component. That glycerol backbone component is going to be glycerol 3-phosphate. Those two things are going to come together and form those storage triglycerides. You can see those right here. So what is this glycerol 3-phosphate? You've seen this before. Glycerol 3-phosphate comes from DHAP, dihydroxyacetone phosphate, which is an intermediate in glycolysis. So remember how we said lipogenesis is stimulated by insulin. So we're all operating right now under an insulin-mediated process. Well, there are other insulin-mediated processes like the insertion of the GLUT4 transporter onto the adipocyte membrane, which brings glucose in from the blood into the adipocyte where it can undergo glycolysis. So we bring our glucose in, we do our first couple steps of glycolysis, we get to our F16BP intermediate, which remember splits into DHAP and glyceraldehyde 3-phosphate. And those things go on to eventually become pyruvate, right, which is the end product of glycolysis. And that can be acted upon by pyruvate dehydrogenase to become acetyl CoA. And this is all stuff we covered in the glycolysis video that you should be familiar with by now, but of course is a good review. But then that DHAP, not only can it turn into that glyceraldehyde 3-phosphate, but it can turn into a glycerol 3-phosphate. That glycerol 3-phosphate is what we're interested in for the purpose of lipogenesis because it's going to become the glycerol backbone of this storage triglyceride. And it'll combine with that acyl CoA. So let's go back to the outline. We first talked about how the fatty acids got into the adipocyte from the blood. That was LPL cleaving the triglycerides out from chylomicrons and VLDLs, turning it into glycerol and fatty acids. Those fatty acids then moved into the adipocyte via those FATPs, those fatty acid transport proteins. Those FATPs were also helpful in our next step, which was conjugating that fatty acid to a CoA, turning it into acyl CoA via the enzyme acyl CoA synthetase. And that acyl CoA was the fatty acid portion of our storage triglyceride, which merged with glycerol 3-phosphate, which was the glycerol backbone portion of that triglyceride. And all those components merged together to form that storage triglyceride. And we said that that glycerol 3-phosphate came from DHAP, which was from glycolysis, which is also going on at the same time, right? Because this is an insulin-mediated process. One thing to note here, there is also a minor pathway for lipogenesis, where acetyl-CoA from glycolysis, that can just turn right into fatty acids, which then will merge into triglycerides. That's the minor pathway. That's unlikely to be tested compared to this mechanism, which we just finished walking through. All right, so that was lipogenesis, the storage of triglycerides. Now, how do we release those energy stores when it's time, like when we are exercising or fasting? That is going to be stimulated by all of those things that were inhibiting lipogenesis. So that's glucagon, epinephrine, norepinephrine, ANP, and BNP. And lipolysis is going to be decreased by insulin. And this is a high yield point because it explains why you can get insulin-induced hypoglycemia. So if you've ever met a patient or known someone personally who has diabetes, who is dependent on insulin, you know one of the feared complications of too much insulin is insulin-induced hypoglycemia. That's where you give too much insulin and all of the sugar from your blood goes rushing into your cells because of the stimulation of the insertion of that GLUT4 transporter, promoting glucose intake into cells. That's a problem because normally in your body, insulin is a signal of higher blood sugar, of a fed state. Your body doesn't know what to do with the fact that you have a lot of insulin going on and your blood sugar is low. So when insulin-induced hypoglycemia, the increased insulin is going to favor glucose use, bringing that glucose in from the bloodstream into the cells, over the use of fatty acids or ketones, because that insulin decreases lipolysis, right? Because your body's thinking, why would I liberate all of my energy stores? I have all this insulin around. I must be in a fed state. I must have plenty of glucose. Insulin reduces lipolysis, and that means that lipolysis is not going to help you out in this situation of insulin-induced hypoglycemia. You can get a partial compensatory response from epinephrine, but it's not enough to bring you back up to baseline. Understanding that point is very high yield. So let's actually talk about that lipolysis. And this is going to be really easy to wrap your head around. If you understand, again, we've got this glycerol backbone and we've got 3 tails. Each step of this lipolysis is just going to be cleaving off one of those tails, each of those tails being a free fatty acid. So the first enzyme is going to be adipocyte triglyceride lipase. That's going to pluck that first fatty acid tail off and form a diglyceride from a triglyceride. That diglyceride is going to have another fatty acid tail plucked off via hormone-sensitive lipase, and that will leave you with a monoglyceride. And just so you're aware, in a lot of books and even test questions, they're going to sort of use hormone-sensitive lipase as the representative enzyme for lipolysis. So if you have to memorize one name, it would be the hormone-sensitive lipase, which turned our diglyceride into a monoglyceride, and then monoglyceride lipase is going to take that monoglyceride, pull off that glycerol backbone, and just leave you with another fatty acid. And all those fatty acids that we liberated can all go out into the bloodstream, again, via that FATP. Those fatty acids in the blood will bind to albumin, which is their carrier. And those can go off to the muscle and the liver for beta oxidation. And we'll talk about beta oxidation in an upcoming video, but that's basically how we actually get ATP energy from these fatty acids. And that beta oxidation can also happen with all of these fatty acids that are here within the adipocytes. Now, this glycerol that we liberated can also go out into the blood. This can travel unbound. It doesn't have to be bound to a carrier like albumin. And this is going to go to the liver for gluconeogenesis. So we can also use this glycerol to help us make more energy in this fasting state. You can see here on our metabolism map, which we are never done with, that glycerol can turn into DHAP, which can run all the way back up the chain, this glycolysis in reverse, aka gluconeogenesis. So back to the outline, the steps of lipolysis. These triglycerides turn into fatty acids and diglycerides via that enzyme adipocyte triglyceride lipase. Then the diglycerides turned into monoglycerides, freeing out another fatty acid tail via that enzyme hormone-sensitive lipase. And remember, hormone-sensitive lipase was sort of our representative enzyme when you're thinking about lipolysis. Then we'll take that monoglyceride and we will split that last fatty acid tail off of the glycerol backbone, and the enzyme that does that is monoglyceride lipase. The fatty acids go out into the blood, bind to albumin, head to the muscle and the liver for beta oxidation, which was that last step we needed to actually turn those free fatty acids in the blood into ATP. We said that beta oxidation could also happen within the adipocytes. And then the glycerol went into the blood, unbound to anything, didn't need any albumin, which went to the liver for gluconeogenesis, another energy source in the fasting state. So let's wrap this up. We've been talking about what goes on in the adipocytes during the fed state versus the fasting state. Remember, the fed state mediated by insulin is going to promote lipogenesis, or the storage of triglycerides, whereas mediators of the fasting state like glucagon, epinephrine, norepinephrine, ANP, BNP, those all instead promoted lipolysis, which is the liberation of fatty acids into the blood from their storage form in the adipocytes. We really focused in on insulin and how it promotes the intake of glucose into these adipocytes via the insertion of the GLUT4 transporter, and how that all helps to feed into the lipogenesis pathway, but how that can lead to problems during insulin-induced hypoglycemia, because insulin inhibits lipolysis. We talked about the steps of lipolysis and how we were basically just plucking individual fatty acid tails off of the storage triglycerides and releasing those into the blood, where they could go off to peripheral tissues and participate in beta oxidation, which is the final step to actually turn these fatty acids into ATP for energy. And that's what we're going to talk about in the next video, so stay tuned. Till then, that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on fatty acid degradation, also known as beta oxidation. So in our video about lipolysis, we talked about how in the fasting state, when your body needs to liberate energy stores, triglycerides, that storage form of fats, can be broken down into free fatty acids and dropped out into the blood. And beta oxidation is the process by which we can actually turn those free fatty acids into energy. So fatty acids will be catabolized via beta oxidation in the mitochondria of the liver and in the muscle and the adipose tissue. And the byproduct of the beta oxidation process is acetyl CoA. And if you take a look at our metabolism map, you'll see our old friend acetyl CoA right here at the center. By turning fatty acids into acetyl CoA and creating acetyl CoA, we are creating an energy source that can feed either into the TCA cycle for energy or into the ketone metabolism pathway. So the goal of beta oxidation is to liberate acetyl CoA from those fatty acids to feed those processes, and then we'll also make FADH2 and NADH along the way. And if you'll remember, those were electron carriers for the electron transport chain, which is the cornerstone of aerobic metabolism and aerobic ATP production. So we said beta oxidation happens in the mitochondria of some of these peripheral tissues. Let's talk first about how we get the fatty acids into the mitochondria, because that's a testable point. So remember from our Intro to Lipids video, we can categorize fatty acids by the number of carbons along their carbon tail. So short chain and medium chain fatty acids, basically any fatty acids fewer than about 12 carbons, can diffuse freely into the mitochondria. Easy enough to wrap your head around. Long chain fatty acids, though, are going to need more of a specialized chaperoning process across that mitochondrial membrane. And that chaperoning process is going to be a carnitine-dependent shuttle. So this is a massive diagram of fatty acid metabolism. And on the left half of the image is fatty acid synthesis, which we're not talking about today. We'll talk about in the next video. We're going to take a look here on the right half of the image, which is focused on fatty acid degradation. Our first step is to take this fatty acid and conjugate it to a CoA group to basically activate it. So this enzyme right here, fatty acyl CoA synthetase, is going to combine this fatty acid with a CoA to form a fatty acyl CoA, and we're going to use up some ATP in that process. So we have our CoA group with the fatty acid attached to it, and you can see here on the diagram, we're going to be able to follow this little cartoon fatty acid along as we play hot potato with it the next couple steps. That acyl CoA is going to go to the outer mitochondrial membrane, and it's going to encounter this enzyme right here, carnitine palmitoyl transferase 1, also known as CPT1. And CPT1 is going to take that fatty acid group from the acyl CoA and pass it to a carnitine, forming an acyl carnitine. So we've taken that fatty acid group off of the acyl CoA, just turning it back into a regular old CoA that can go back and join another reaction. And now we have our carnitine that's been loaded up with that fatty acid, forming an acyl carnitine. And that carnitine is the fatty acid's chaperone into the mitochondria. And it'll enter through this transporter here, the carnitine acylcarnitine translocase. So now with the help of that carnitine, we've brought that fatty acid group into the mitochondrial matrix. And now that we're in, we don't need that carnitine chaperone anymore, so we can reunite the fatty acid group with the CoA to reform acyl CoA inside the mitochondria. And just like how CPT1 facilitated the unloading of the acyl CoA, an enzyme called CPT2, carnitine palmitoyl transferase 2, is going to facilitate the reloading of that fatty acid group back onto a CoA inside the mitochondrial matrix. Now that we have acyl CoA in the mitochondria, we can undergo beta oxidation, which we'll talk about the specifics of in a second. In the meantime, that carnitine that we freed out can exit the mitochondria via that same carnitine acylcarnitine translocase, and it's been recycled to chaperone another fatty acid and restart the cycle again. So let's go back to our outline and rehash how those long-chain fatty acids make it into the mitochondrial matrix. First, that fatty acid is going to be conjugated to a CoA group, forming a fatty acyl CoA. That was via the enzyme fatty acyl CoA synthetase in the outer mitochondrial membrane in cytoplasm. The next step was to unite that fatty acid group with its carnitine chaperone. So the enzyme carnitine palmitoyl transferase 1, or CPT1, transferred that fatty acid group from the acyl CoA to form acylcarnitine. And that CPT1 enzyme is the rate-limiting step of fatty acid catabolism. And you know, whenever we see a rate-limiting step in biochemistry, it's time to think about how those steps are going to be regulated. In this case, the regulation is pretty self-explanatory. It is going to be increased by glucagon. Glucagon being our signal of the fasting state, telling your body that it's time to run a catabolic process to free up some energy. So the action of this step is also going to be decreased by insulin. It's also going to be decreased by malonyl CoA. And malonyl CoA right here is an intermediate in the fatty acid synthesis pathway, aka the exact opposite pathway to the one that we're running right now. So if we have more fatty acid synthesis, we of course are going to shut down fatty acid degradation. All right, so that was CPT1, which took the fatty acid off of the acyl CoA and formed acylcarnitine. And now that acylcarnitine can go into the mitochondria through the carnitine acylcarnitine translocase. Once that acylcarnitine was inside, that fatty acid group didn't need that carnitine chaperone anymore. So that fatty acid group could be removed from the acylcarnitine and put onto a CoA to reform acyl CoA via the enzyme carnitine palmitoyl transferase 2, or CPT2. That carnitine could be recycled out to facilitate the transfer of another long-chain fatty acid, and then we had our acyl CoA group finally now inside the mitochondrial matrix, where it's ready to go ahead and start beta oxidation. So now we actually have that fatty acid in the form of fatty acyl CoA inside the mitochondrial matrix, ready to be broken down for energy via beta oxidation. And we can go into as much detail on beta oxidation as we want here, but if your test is tomorrow, in a nutshell, what you should understand about beta oxidation is that beta oxidation involves cycles of fatty acid chain cleavage at the 2-carbon position of a fatty acyl CoA. That cleavage is going to be conducted via an oxidation reaction, and that oxidation reaction is happening at the beta carbon position, which is a fancy word for that 2-carbon position. So let's take a look here. This is our beta oxidation pathway. This is our starting fatty acyl CoA. So here's our CoA group, and then we've got this long fatty acid tail. And this R group is just representing a fatty acid tail of any length you want. Now we're going to want to know how to identify the beta carbon for beta oxidation. To identify the beta carbon, you find the carbonyl group and you start with carbon 0 right at the center. From there, you'll count the alpha carbon and then the beta carbon. So that beta carbon is 2 carbons away from the center of that carbonyl group. And so you can see here's our starting acyl CoA. We're going to go through a series of reactions, and we're basically going to cleave this bond between the alpha and the beta carbon. We're going to do that by oxidizing the beta carbon. And so you can see after all of these reactions, we've split between the alpha and the beta carbon. We have oxidized that beta carbon. We put a CoA group on the end of it. And by cleaving at that location right there between the alpha and beta carbon, we shortened this acyl CoA by 2 carbons. And wouldn't you know it, this 2-carbon group that we cleaved off the end of our acyl CoA is the structure of acetyl CoA. So we freed out an acetyl CoA and we are left with an acyl CoA that has been shortened by 2 carbons. Those 2 carbons that went towards making that acetyl CoA. And the longer the fatty acid, the more cycles it can go through and the more acetyl CoAs you'll get out of it. And you can keep running that beta oxidation and pulling off 2 carbon groups until you get to a chain with either 4 carbons left if it started with an even number of carbons on the chain, or 3 carbons left if it started with an odd number of carbons on the chain. We'll talk about that termination process in a second. Now, as you can see, there are 4 enzymatic steps of the actual beta oxidation process. The first step is an oxidation step mediated by acyl CoA dehydrogenase, and that'll give you an FADH2 byproduct. That FADH2 can go off to the electron transport chain to participate in ATP synthesis. The second step is a hydration step mediated by enoyl CoA hydratase. The third step is another oxidation step mediated by 3L-hydroxyacyl CoA dehydrogenase. That's going to form an NADH as a byproduct, which can also go off to join the electron transport chain. And then our fourth step is a thiolytic cleavage, which is a fancy word for shoving a CoA group on the end of our chain. And that's going to be mediated by beta ketothiolase. I'm going to say memorizing these exact enzyme names is less important than understanding the overall inputs and outputs of beta oxidation. So we put in a fatty acyl CoA, and remember we got out an acyl CoA that had been shortened by 2 carbons, which could go ahead and re-enter beta oxidation until the chain got as short as possible. And remember, those 2 carbons that we shortened it by were liberated in the form of a 2-carbon acetyl CoA, which could then go on to join energy-producing reactions like the TCA cycle or ketogenesis. And that was actually how we got energy out of this process. We also got energy out via the liberation of FADH2 and NADH each cycle, and those things, remember, could go on to join the electron transport chain. If the fatty acid we started with had an even number of carbons, we'll keep running beta oxidation until there are 4 carbons left on the chain. And we'll split that final 4-carbon chain into 2 2-carbon acetyl CoAs. And those acetyl CoAs are free to join the TCA cycle or ketone metabolism, or go on to be part of the citrate shuttle, which we will talk about in the next video on fatty acid synthesis. However, if that fatty acid that we started with had an odd number of carbons, we'll keep running beta oxidation until we're left with a 3-carbon molecule, and that 3-carbon molecule is called propionyl CoA. Back to our metabolism map, if you zoom in on the bottom right down here, you'll remember from our lessons on 1-carbon metabolism that odd chain fatty acids are one of the things that feed in to become propionyl CoA. Propionyl CoA continues on down further reactions till it eventually becomes succinyl CoA, that TCA cycle intermediate. So that's how beta oxidation runs in the mitochondria. However, you'll notice we talked about short chain fatty acids, medium chain fatty acids, and long chain fatty acids going to the mitochondria. We didn't talk about what happens with very long chain fatty acids. And way back in chapter 1 on cell biology, we did a video on the peroxisome, and we said that beta oxidation for very long chain fatty acids begins in the peroxisomes, and those very long chain fatty acids will go through cycles and cycles of beta oxidation until they are short enough that they are only 8 carbons long. They form an octanyl CoA. Once they're 8 carbons long, they can go into the mitochondria and finish beta oxidation there. Beta oxidation in the peroxisome is similar but not the same to how the process runs in the mitochondria. The important thing to know about beta oxidation in the peroxisome is that it is coupled to the production of hydrogen peroxide, which is a toxic metabolite. And that's why we don't just run this all the way in the peroxisome, because we want to minimize the production of this toxic metabolite. And remember in the video on the peroxisome, I told you about this disorder called adrenoleukodystrophy, which was a defect in the import of very long-chain fatty acids into the peroxisome. That meant that those very long-chain fatty acids couldn't undergo beta oxidation. We got a buildup of those very long-chain fatty acids with no way to break them down. And so those very long-chain fatty acids would build up either in the adrenal glands, causing adrenal insufficiency, or in the white matter, causing a decrease in cognition. You can go ahead and check out the video on the peroxisome for more details on that. So let's sum things up. We talked about beta oxidation, which was how we actually got energy from free fatty acids. First, we talked about how we got those free fatty acids into the mitochondrial matrix. Short and medium chain fatty acids could just diffuse freely. Long chain fatty acids depended on a carnitine-dependent shuttle, and very long chain fatty acids actually had to undergo a slightly different form of beta oxidation in the peroxisomes until they were short enough to join the mitochondria. The process of beta oxidation itself involved starting with a fatty acyl CoA and oxidizing it at that beta carbon or that 2-carbon position, which would cleave off those 2 carbons in the form of an acetyl CoA. And in the process, we freed out FADH2 and NADH. That acetyl CoA joined the TCA cycle or ketogenesis, which were energy-producing pathways, which is how we got energy out of this. And those FADH2 and NADH molecules could also join the electron transport chain for further ATP production. That remaining acyl CoA that had been shortened by 2 carbons could re-enter the beta oxidation cycle until it got short enough that we terminated the chain, either by splitting it into 2 acetyl CoAs for even chains, or by taking the 3-carbon remaining propionyl CoA and feeding that into the TCA cycle as well. And that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on disorders of fatty acid metabolism. We're going to be talking about some inborn errors of fatty acid degradation. If you're already familiar with the fatty acid degradation and beta oxidation video and material, this is going to come very easily to you. Depending on the erroneous step, you'll get buildup of different metabolic intermediates. However, the highest yield disorders, which are systemic primary carnitine deficiency, MCAD deficiency, and CPT2 deficiency, have a lot of similar clinical features, and you can think of them together as a unit for the most part. Let's start by talking about systemic primary carnitine deficiency, right? As the name suggests, it is a primary carnitine deficiency, meaning the chief problem is we don't have enough carnitine in the cells. And if you remember the function of carnitine within the fatty acid degradation pathway, carnitine was our chaperone that allowed this acyl group to get across the inner mitochondrial membrane. No carnitine means no long-chain fatty acid mitochondrial import via that carnitine shuttle. Remember, we're specifying long-chain fatty acids here because that carnitine shuttle is only required for long-chain fatty acids to get into the mitochondrial matrix. Short and medium chains could just diffuse freely. If we don't have that carnitine chaperone across the membrane for the long-chain fatty acids, we're going to end up with a buildup of long-chain fatty acids in the cytosol. And these long-chain fatty acids in the cytosol are toxic. And specifically, the tissues where these like to accumulate are the liver, the cardiac muscle, and the skeletal muscle. One thing I want to emphasize, a primary carnitine deficiency is just that. It is a deficiency in carnitine. There is another disorder called CACT deficiency. That's a deficiency in this transporter here. CACT deficiency is not commonly tested on the Step 1 exam. Just in case you encounter that in your reading, CACT deficiency is not the same as a primary carnitine deficiency, but because in essence they're both interfering with the same step, you're going to have a very similar clinical presentation. All right, the next disorder we'll talk about is medium chain acyl CoA dehydrogenase deficiency, or MCAD deficiency. As this name suggests, we're going to have a deficiency in our medium chain acyl CoA dehydrogenase, which was involved in fatty acid beta oxidation of medium chain fatty acids. Right? So remember, once we had our acyl CoAs in the mitochondrial matrix, they could be acted upon by acyl CoA dehydrogenases, which were sort of the representative enzymes of the beta oxidation process. In MCAD deficiency, we're going to knock that out for medium chain fatty acids. If we can't break down those medium chain fatty acids via beta oxidation, we're going to get a buildup of medium chain fatty acyl CoAs in the liver. So when we knock out this step, we get a buildup of the upstream intermediate, those medium chain acyl CoAs. And then the last disorder we'll talk about is called carnitine palmitoyltransferase 2 deficiency, or CPT2 deficiency. CPT2 was in charge of actually transferring that acyl group off of the acyl carnitine back onto CoA to reform the acyl CoA that could undergo that beta oxidation. In a CPT2 deficiency, that step is not functional. So we can't unload these acyl groups off of the acyl carnitines. We get a buildup of these acyl carnitines. And again, this is going to be specific to long-chain acyl carnitines because only long-chain fatty acids require the carnitine shuttle to get into the mitochondrial matrix. So a decrease in CPT2 activity causes a buildup of long-chain acylcarnitines, specifically in the liver, in the heart, and in the skeletal muscle. So in this case, the problem isn't actually with getting carnitine into the cells. We just can't take the acyl group off of the carnitine. So all the carnitine is sequestered as acylcarnitine, and you get a decrease in the total and free carnitine. All right, so a lot of these disorders are going to look pretty similar because they're all sort of happening right next to each other in this degradation pathway. And there's a lot of overlap in the tissues they affect. So in all three of these, you're going to see some common diagnostic and clinical features like weakness, hypotonia, and liver dysfunction. So examples of liver dysfunction that you might see in a vignette are hepatic encephalopathy. You might see hyperammonemia, as well as elevated liver function enzymes like aminotransferases, AST, ALT. However, I always found all of those incredibly vague, not at all helpful in narrowing the differential. If you take one single thing away from this video, if you don't even understand any of the mechanisms or what have you, remember that a disorder of fatty acid metabolism is going to have a hypoketotic hypoglycemia, as well as a lactic acidosis. This is because we are interfering with beta oxidation and ketogenesis inside the mitochondria. So take a look. Here's our beta oxidation step. Here's our ketone metabolism step, right? Because beta oxidation liberates out acetyl-CoA, which can go on to form ketones. We'll talk more about that in the next video, but you could see any one of these deficiencies is interfering upstream from beta oxidation and ketone metabolism. So normally when you're hypoglycemic, you should see an increase in ketones in the bloodstream. Your body should be trying to compensate for that low blood sugar. But in this family of deficiencies, your body cannot run those pathways. So in these vignettes, they'll do a fingerstick glucose or some sort of glucose measurement. That'll be low. And then when they do a ketone measurement, those ketones will also be low. That's very unique to this family of disorders. And then a couple other clinical features that you'll see in primary carnitine deficiencies and CPT2 deficiency are some additional skeletal muscle and cardiac findings like skeletal myopathy, rhabdo, and dilated cardiomyopathy. MCAD deficiency was primarily affecting the liver, and that's where those symptoms are sort of going to stay, as opposed to these other two, which had buildup in a wider variety of tissues. So I'll tell you the way that these vignettes at least used to go for me. Maybe they'll go for you the same way. I would read this very vague vignette about a child. Many times they were a home birth or something because a lot of these disorders are caught on a newborn screen. And the child has a bunch of these very vague symptoms like this weakness, this hypotonia. Maybe they've got poor feeding. Maybe they have altered mental status. Usually for questions about these disorders, I would get to the end of the history section and not have a very strong differential. But in these questions, almost always, they give you labs. When you see hypoketotic hypoglycemia, you don't even have to look for the lactic acidosis. That finding should be screaming at you one of these disorders of fatty acid metabolism. It's very unlikely that they're going to put multiple of these in as answer choices. Most commonly, they'll be asking about an MCAD deficiency. If you see some of these additional skeletal and cardiac symptoms, you'll be thinking about these other disorders. Now, the management is going to vary slightly based on the disorder. So for a systemic primary carnitine deficiency, as well as all of these disorders of fatty acid metabolism, you're going to want to avoid fasting, right? Hypoketotic hypoglycemia is not a state you want to be in. It's low blood sugar. Your body has no way to compensate for it. The next most obvious intervention is going to be oral carnitine supplementation. And in an acute setting, this is going to look like IV carnitine with dextrose, right? You need to give them sugar to address the low blood sugar. You need to give them carnitine to address the underlying problem. In an MCAD deficiency, again, avoid fasting. Here you're looking for a high carb and a low fat diet. We can't do beta oxidation. We're going to want to rely on glucose instead of fatty acids for our energy. In the acute setting, again, you're going to give IV dextrose, basically just giving them sugar. In a CPT2 deficiency, again, same theme, avoid fasting. Here you're going to have a diet that's high in medium chain fatty acids and high in carbs and low in triglycerides. Remember, medium chain fatty acids don't require CPT2 to get into the mitochondria. They can just diffuse freely. So that's an additional energy source tool in the arsenal we can use for CPT2 deficiency. along with carbs for energy. So let's wrap it up. We've been talking about disorders of fatty acid metabolism, which are inborn errors in fatty acid degradation. In systemic primary carnitine deficiency, we had a decrease in systemic cellular carnitine uptake. This caused a toxic buildup of long-chain fatty acids in the cytosol of liver, cardiac, and skeletal tissues. In an MCAD deficiency, we had a decrease in medium-chain fatty acid beta oxidation due to a decrease in that MCAD enzyme. This led to a buildup of medium-chain fatty acyl CoAs in the liver. In the CPT2 deficiency, we had a decrease in CPT2 activity, which led to a buildup of long-chain acyl carnitines in the liver, the heart, and the skeletal muscle. Lots of vague symptoms like weakness and hypotonia, liver dysfunction common to all of them. However, like we really hit home in this video, hypoketotic hypoglycemia is what you're looking for on labs. And a couple of extra skeletal muscle and cardiac findings on primary carnitine and CPT2 deficiencies, not in MCAD deficiency. And different dietary modifications are done for management of each of these different disorders, but the common theme is to avoid fasting, avoid the pits of hypoglycemia. You won't get into that hypoketotic hypoglycemia crisis. And we're going to talk a lot more about ketones in the next video coming up, so stay tuned. Till then, that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige. Thank you so much for joining me for our lesson on fatty acid synthesis. As we discussed in our introductory video to this chapter, there are essential fatty acids and non-essential fatty acids. So essential fatty acids are ones that we must take in from our diet. But remember, there's this whole other category of non-essential fatty acids that we can actually make ourselves. We can synthesize fatty acids from excess glucose, and that's what we're going to be talking about in this video. So the only fatty acid synthesized de novo in humans is palmitic acid. And palmitic acid is 16 carbons long, and this is going to be the primary end product for our fatty acid synthesis pathway. Once you have the 16 carbon palmitic acid, your body can do other elongations and manipulations to it, but this palmitic acid is the primary end product you're going to be focused on for now. It's going to take place in the cytoplasm of the liver, the adipose, as well as lactating mammary glands. And the reactants that we're going to feed into this reaction to get that palmitic acid are 8-acetyl-CoA, 14 NADPH, and 7 ATP for every palmitic acid chain. And you're unlikely to be tested on those numbers, just understand the reactants and the products. All right, so here's the diagram that we're going to be working with for this video. In the video on beta oxidation, we already talked about the right half of this diagram, which talked about fatty acid degradation. Now we're focused on the synthetic pathway. And we're going to start with acetyl-CoA over here in the mitochondrial matrix and work our way out until we end up with a final fatty acid that palmitate out in the cytoplasm of those cells. But let's talk as a refresher first as to how that acetyl-CoA gets into the mitochondrial matrix. Remember, fatty acids are going to be made from excess glucose. And following glucose down glycolysis, which we've done many times by now in this biochemistry unit, we break glucose down into pyruvate in the cytoplasm of cells. Pyruvate, remember, is acted upon by the pyruvate dehydrogenase complex and becomes acetyl-CoA inside the mitochondrial matrix. So we turned our glucose into pyruvate via glycolysis, and then our pyruvate went into the mitochondria and became acetyl-CoA via the pyruvate dehydrogenase complex. And that's where we picked it up down here in our diagram. Now we're going to use something called the citrate shuttle to get that acetyl-CoA from the mitochondria back out into the cytoplasm. So it's confusing because we just brought that pyruvate into the mitochondrial matrix to turn it into the acetyl-CoA, and now that it's the acetyl-CoA, we're going to put it back out into the cytoplasm if we're going to use that acetyl-CoA for fatty acid synthesis. So in the case of the TCA cycle, we have our acetyl-CoA in the mitochondrial matrix, and we just run the TCA cycle right from there. But today we're talking about fatty acid synthesis. So we got to get that acetyl-CoA back out into the cytoplasm. And we're going to use that citrate shuttle we were just alluding to. Acetyl-CoA is going to merge with oxaloacetate to form citrate. That's the same as the first step of the TCA cycle. You're familiar with that already. But instead of running on through the rest of the TCA cycle from citrate, instead we're going to take that citrate and we are going to export it out of the mitochondria. And then we're going to split it back into its component parts. So we combine those two, turn them into citrate, brought that out, and then this enzyme right here, ATP citrate lyase, is going to be in charge of cleaving that citrate apart into those same two starting products, the acetyl CoA and the oxaloacetate. So we just used citrate as a neat little package to bring that acetyl CoA across that membrane. All right, so that's our citrate shuttle. We've got the acetyl CoA and the oxaloacetate, which turns into citrate, goes out to the cytoplasm, and then splits back out into its component parts via the ATP citrate lyase. Our next step is to turn acetyl CoA into malonyl CoA. Now, acetyl CoA is 2 carbons long, malonyl CoA is 3 carbons long. So the enzyme that's going to turn our 2 carbon into a 3 carbon is going to be a carboxylase, which adds another carbon. And that enzyme is called acetyl CoA carboxylase, and it's got a couple of cofactors that are required, namely biotin or vitamin B7, ATP, CO2. You can see that on the diagram here, where now that we're out in the cytoplasm, the acetyl CoA is going to be carboxylated via this acetyl CoA carboxylase to turn into malonyl CoA. We've got our B7, our CO2, and we're also going to require ATP. And that ATP is going to phosphorylate our acetyl CoA carboxylase, and that is a regulatory step. So that'll be upregulated by insulin, high glucose, ATP, and citrate. All indications of the fed state, saying that, okay, we have enough energy, we have extra glucose to spare, let's turn it into some fatty acids. Fatty acid synthesis is going to be decreased by glucagon, low glucose, catecholamines like epinephrine, norepinephrine, indications of exercise, a fight or flight state, AMP, which is a signal of a low energy state, and then acyl CoA and palmitoyl CoA. Those basically just telling us we've already got plenty of fatty acids present and synthesized in the body. All right, now that we've got our malonyl CoA, we're actually going to do the synthetic steps to turn this 3-carbon malonyl CoA into our 16-carbon palmitate. So the next couple things that we're going to talk about, they're not really shown on this diagram too much, but they're all going to be happening right here, starting from malonyl CoA and turning them into the palmitate. What's going to happen is an enzyme called fatty acid synthase is going to add 2 carbon units to fatty acid chains until we get to 16 carbons. So this should be striking a parallel to you with beta oxidation, where in beta oxidation we were cleaving off 2 carbon units with each cycle. Now we have the anabolic equivalent of that, where we're going to be adding on 2 carbon units to the chain with each cycle. So to start this chain, we're going to take this malonyl CoA and we're going to combine it with acetyl CoA. Acetyl CoA is 2 carbons, malonyl CoA is 3 carbons. So we're going to take our 2 carbons and our 3 carbons and we're going to smash them together, basically. And a series of reactions is going to take place, one of which is a decarboxylation. So that's going to take 1 carbon out of the picture. Then we'll do some other things like hydration and reduction with NADPH. So this is where that NADPH comes in. NADPH, remember, from the pentose phosphate pathway, or the HMP shunt, which we already did a video discussing. And so when we take our 2 carbon and our 3 carbon, we mash them together and we decarboxylate them. So we take one of those carbons out and we do a whole bunch of other stuff with it. We end up with a 4 carbon fatty acid bound to fatty acid synthase, which is the synthetic enzyme that's mediating this whole thing. So that was our initiation step, and so now we've got this 4-carbon chain bound to the enzyme, and we can keep elongating via similar steps. So we're going to keep doing the same thing that we did before with the malonyl CoA. Now we've got our 4-carbon chain. We're going to add our 3-carbon malonyl CoA, and then we'll decarboxylate it, which knocks another carbon off, do these other steps here, and then at the end of the next cycle, it'll be 6 carbons. And then we'll take another malonyl CoA and add it on and decarboxylate and do all the other stuff, and then it'll be 8 carbons. So you keep going and adding on 2 carbons each time, right? So of the 3 carbons in the malonyl CoA, only 2 of them are getting added onto the chain each time, because one of the steps is a decarboxylation step. And so because malonyl CoA is our building block for all of these elongation steps, the concentration of malonyl CoA is the rate limiting factor in this elongation reaction. And like I said, we keep doing this all the way up until we get to 16 carbons, which is palmitate. And so once you get to that 16 carbon palmitic acid end product, technically the fatty acid synthesis pathway, as we've talked about it, ends. However, you can extend the chain longer than 16 carbons. It's just a different process with different enzymes. The details of it are unlikely to be tested, but in a similar way to what we've been talking about before, we can use elongase enzymes this time, different from the FA synthase enzyme, but similar concept, to add malonyl CoA groups on and continue to elongate that chain up to 18 to 24 carbons and even beyond. These chains can get up to 40 carbons long in places like the skin, the brain, sperm, and the retina. And we're still discovering the functions of these ultra long 40 carbon fatty acid chains in some of those tissues. And then, as we sort of talked about in some of these previous videos in this chapter, metabolic reactions with fatty acids require activation into fatty acyl CoAs. Now, here's a sketch of palmitic acid, and as you can see, it is saturated, right? We've got no double bonds in there. Another modification to this palmitic acid that your body can do is desaturation, where you add in double bonds. Specifically, this is happening at the 5, 6, or 9 position from the carboxyl end, and there is no insertion beyond C9. So remember, this is your carboxyl group, and this is the first carbon, and so you can insert double bonds, for example, at this position, right? Anywhere at the 5, 6, or 9, but past the 9 position, you cannot insert double bonds. Your body doesn't know how. But as you know, we have things like omega-3 fatty acids, which have a double bond 3 carbons away from the distal end of this fatty acid. And that's why we have essential fatty acids. These long chain unsaturated fatty acids with double bonds near that omega end, out past where your body's natural desaturation reactions can touch them, are essential. And those are things like our examples of linoleic acid, which was in omega-6, and the alpha-linolenic acid, which was in omega-3. So the fact that we have to eat them and can't make them ourselves makes them essential. But essential fatty acids can be desaturated and elongated just like non-essential fatty acids. All right, so let's wrap it up. In this video, we've been talking about fatty acid synthesis from excess glucose, specifically the formation of palmitate or palmitic acid, that 16 carbon unsaturated fatty acid. We took glucose, we put it through glycolysis and the pyruvate dehydrogenase complex, eventually ended up with acetyl-CoA in the mitochondrial matrix. Fatty acid synthesis, though, happens out in the cytoplasm, so we have to bring that acetyl-CoA back out to the cytoplasm. We did that via the citrate shuttle. Once we had that acetyl-CoA in the cytoplasm, we could turn it into malonyl-CoA via acetyl-CoA carboxylase. This was our regulatory step that was increased by signs of the fed state and decreased by signs of the fasting state. And then once we had that malonyl-CoA building block made and ready, we could use it to initiate the chain and then further elongate the chain, adding on 2 carbons with each cycle until we reach that 16 carbon palmitate. And then if we wanted to get it any longer than that, we had to use special elongase enzymes through a slightly different process. And then we talked about desaturation and how we can further modify some of these fatty acids by adding in double bonds, but we cannot add double bonds past the 9 carbon position. And that's why these long chain unsaturated fatty acids like omega-3s and omega-6s are essential fatty acids. And that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on ketone synthesis and oxidation. So we've been talking a lot about different energy sources in this chapter, and we have reached one of our final energy sources that we have yet to discuss, which are ketones. These are a water-soluble energy source, and they are released in periods of prolonged glucose deficiency. Ketones are released in periods of starvation, late stage, long-term fasting. However, like we've discussed in previous videos throughout this whole biochemistry unit, there are other more specific scenarios where you might see elevated levels of ketones in the blood. That is diabetic ketoacidosis and alcoholic ketoacidosis, and we're going to review the mechanisms for those in this video as well. Ketones are produced in the liver by hepatic mitochondria, and then those ketones are going to be exported from the liver and used by the mitochondria of extrahepatic tissues out in the periphery, so things like the heart, the brain, the renal cortex, and the skeletal muscle. However, like we said, these are used by mitochondria within those cells. That means that things like red blood cells that do not have mitochondria cannot use ketones as an energy source. They're still going to rely on glucose, specifically glycolysis, which does not require a mitochondria. And there are 3 different sort of flavors of ketones. The first is acetoacetate, the second is beta-hydroxybutyrate, and the third one is acetone. And acetone is kind of the breakdown product of those other first two types of ketones. We're going to kick off our discussion on ketogenesis by taking a look at our diagram down here. So I want to preface this discussion by saying, please do not, under any circumstance, memorize all of these different steps and the organic chemistry arrow pushing stuff. This is really just to give you a visual representation of ketogenesis. So down here we have our three end products, the three different types of ketones. We have our acetoacetate here, and this is sort of the first one that's going to be made in this process. And then based on its structure, you can kind of see how we would get to beta-hydroxybutyrate or acetone by modifying this structure. As you can see, I've sort of color-coded these acetyl-CoAs, and it's actually going to take 3 acetyl-CoAs to form this final acetoacetate. So let's walk through our steps here. We're going to take 2 acetyl-CoAs and turn them into acetoacetyl-CoA, basically merge them together and spit out one of these CoA groups, and that'll leave you with acetoacetyl-CoA. And the enzyme that mediated that step is going to be called thiolase. And that reaction is a reversible reaction, so we're going to come back to that a little later, because that's not the last we're going to see of this thiolase enzyme. Once we have that acetoacetyl-CoA, you can already see the skeleton of our acetoacetate starting to come together. Our next step is going to be to turn this acetoacetyl-CoA into HMG CoA via the enzyme HMG CoA synthase. And these are going to look familiar to you. Remember that HMG CoA, we saw that before when we talked about cholesterol synthesis. One thing I really want to emphasize though is that this HMG CoA synthase is mitochondrial, okay? It is not cytosolic like the HMG CoA synthase that we saw in cholesterol synthesis. And so mitochondrial HMG CoA synthase is going to take this acetoacetyl-CoA and it's going to feed some water and another acetyl-CoA into this reaction. So now we've added this other carboxylic acid group onto one end of our sort of growing chain. And then to get to our acetoacetate, all we have to do is cleave off one of our original acetyl-CoAs. That step is going to be mediated by HMG CoA lyase, and it'll cleave this bond right here and pop off this acetyl-CoA, leaving us with acetoacetate, which was one of our ketones. Now acetoacetate can take one of two paths. It can either turn into beta-hydroxybutyrate with a little bit of help from NADH and this enzyme beta-hydroxybutyrate dehydrogenase, or it can undergo spontaneous decarboxylation and turn into acetone. So let's go back to the outline. Again, don't focus too much on that arrow pushing. You don't have to be able to draw any of that. It was just a visual aid, just to make sure that you understand the steps. We're going to combine these two acetyl-CoAs together using thiolase as our catalyst and create acetoacetyl-CoA. We're going to take that acetoacetyl-CoA, we're going to throw another acetyl-CoA on there to create HMG CoA. The enzyme that's going to mediate that is mitochondrial HMG CoA synthase, which was not the same as the HMG CoA synthase in the cytosol that we saw involved in cholesterol synthesis. Our HMG CoA lyase enzyme is going to take that HMG CoA, break off one of the acetyl-CoAs, and leave us with acetoacetate, which was our first of our ketones. Then once we had that acetoacetate, we could make our two other ketones, either by reducing acetoacetate into beta-hydroxybutyrate or by spontaneous decarboxylation into acetone. And acetone is going to be exhaled. And acetone in the breath gives off what they describe as a fruity odor, very commonly in test questions. In practice, it's more like nail polish remover. And here's our little cartoon schematic, right? So we've got our acetyl-CoA, which came from fatty acids and amino acids, going down through all the steps we just discussed. You can see we've got our acetoacetate and our beta-hydroxybutyrate. These are going to go out into the bloodstream and eventually out to the peripheral cells. But then remember, acetoacetate, when it's in the blood, can spontaneously decarboxylate, turn into acetone, and that acetone will be expired or exhaled in the breath, giving it a fruity odor or a nail polish remover odor. So let's talk about ketoacidosis, right? The state of an excess of ketones in the blood. We've talked about these mechanisms in previous videos throughout the unit, so this should be more of a review for you if you've been following through chronologically. In the case of fasting, as well as the case of diabetic ketoacidosis, or DKA, what's going to happen is oxaloacetate, which is a TCA cycle intermediate, is going to be depleted for gluconeogenesis. And this is going to cause a buildup of acetyl-CoA which will divert over to ketogenesis. We're going to take a look at our metabolism map. We're going to start by zooming in right over here. And here you can see in the light blue, we are looking at gluconeogenesis. So in the fasting state, as well as diabetic ketoacidosis, gluconeogenesis is ramped up and up and up, either due to a lack of glucose itself in the fasting state or due to a lack of insulin allowing that glucose to actually get into those peripheral cells. Either way, your cells end up just trying to make their own glucose. The starting reactant for gluconeogenesis is oxaloacetate, right? Remember, we turned that oxaloacetate into malate. We brought that malate out of the mitochondria, turned it back into oxaloacetate, and then we fed it back in to go up and run glycolysis in reverse, running it all the way up the chain until we created glucose. That was our gluconeogenesis pathway. So when gluconeogenesis is ramped up and up and up, we are going to deplete our oxaloacetate in order to run that pathway. What that means is there's not enough oxaloacetate to combine with acetyl CoA to start the TCA cycle. So we're going to get a buildup of acetyl CoA, and that acetyl CoA can't go into the TCA cycle. Instead, it's going to find a detour route, and it's coming out this way down the ketone synthesis pathway. And that's how you end up with ketoacidosis in the fasting state, as well as DKA, diabetic ketoacidosis. Alcoholic ketoacidosis is similar, but not quite the same. It's similar because in the end, it's going to be a depletion of oxaloacetate that leads to a buildup of acetyl CoA that then diverts into ketogenesis, just like we finished talking about. But that oxaloacetate depletion is not from running gluconeogenesis in this case. It's because alcohol is going to increase the amount of NADH in the bloodstream, which promotes the conversion of oxaloacetate to malate. Now, this diagram is from our video on the malate aspartate shuttle, and these are topics that we talked about in that video, as well as the video on ethanol metabolism. But one of the things that ethanol did was it promoted the formation of NADH in the cytosol. Now, this had multiple end effects, including lactic acidosis and increased fatty acid synthesis and things like that. But for this video, just understand that that increased NADH is going to favor this forward reaction, which was part of the malate aspartate shuttle, depleting oxaloacetate and turning it into malate. And that oxaloacetate depletion has the exact same effect it had when we talked about the fasting state and DKA. Causes a buildup of acetyl CoA that shunts over to the ketone synthesis pathway. Both of those mechanisms are very high yield to understand. They love to test those because it really tests your understanding of the integration of a lot of these different themes in biochemistry. Patients with ketoacidosis are going to have a couple of signs and symptoms. Polyuria and polydipsia is very classic, especially for diabetic ketoacidosis. That's urinating a lot and drinking a lot. They also classically like to describe that fruity breath odor, which is that acetone on the breath. Then you might also see things like nausea, vomiting, abdominal pain, and altered mental status. From a lab's perspective, you're going to see an anion gap metabolic acidosis. And that's because we're getting a buildup of these keto acids. Those acids are going to be buffered by bicarbonate in the blood, and so it's using up that bicarb that's in the blood to buffer those acids. And ketones are one of those things that cause an anion gap. They're not one of the anions that is accounted for in our lab work. The other thing is all that polyuria and vomiting and all those can cause dehydration. And that can lead to electrolyte imbalances like hyponatremia, hypokalemia, hypophosphatemia. You get the picture. To test for ketones outright, there are two different tests you can do. You can do a urine test or you can do a blood test. The urine test is called the urine nitroprusside ketone test. And that's testing for acetoacetate versus the blood test that you're ordering in the hospital more commonly tests for beta hydroxybutyrate. So just be aware, there's no such thing as just ordering ketones. To manage ketoacidosis, you're giving insulin and glucose. That glucose often in the form of D5 normal saline. And there's a lot of other considerations you have to take into account, like fluid resuscitation and all that. But in terms of step one, the answer they like is glucose and insulin. You give the glucose and you give the insulin to actually shift all of that glucose into the cells. Now, in alcoholic ketoacidosis, you're going to precede that glucose and insulin with thiamine administration. And that's going to help to prevent Wernicke encephalopathy. So Wernicke encephalopathy is this triad of ophthalmoplegia and confusion and ataxia. It's caused by a thiamine deficiency, right? Thiamine, vitamin B1, is a really important cofactor for the TCA cycle. Many patients with alcoholic ketoacidosis might be long-term chronic alcohol users, and those patients are commonly deficient in thiamine. So if these patients are deficient in thiamine, that TCA cycle cofactor, and you give them insulin and glucose, they're not going to actually be able to send that glucose through the TCA cycle because they're missing that cofactor. So before you give them that, you have to make sure that they have enough thiamine to actually be able to utilize the insulin and the glucose. Otherwise, they end up with that Wernicke encephalopathy due to thiamine deficiency. All right, finally, let's talk about ketogenolysis, how we actually break down those ketones into energy. So over here, we had our ketogenesis in the liver, and then remember, we exported those ketones out to the peripheral tissues. Now we're in the peripheral tissue, our example here being muscle, and we're trying to actually use those ketones. First thing we're going to do, if we made beta-hydroxybutyrate, we're going to turn it back into acetoacetate using the same enzyme that formed it, that beta-hydroxybutyrate dehydrogenase. Our next step is going to use a new enzyme, and it's going to take that acetoacetate, and it's actually going to combine it with succinyl-CoA from the TCA cycle. It's a TCA cycle intermediate. We'll combine that acetoacetate and that succinyl-CoA from the TCA cycle into acetoacetyl-CoA via the enzyme thiophorase. And that acetoacetyl-CoA is the same acetoacetyl-CoA we talked about in the ketogenesis pathway. So now we're sort of stepping our way backwards. And if you remember, our acetoacetyl-CoA was just what we got when we basically combined two acetyl-CoAs. So via the enzyme thiolase, which we're already familiar with, remember that thiolase ran a reversible reaction between acetoacetyl-CoA and acetyl-CoA. That thiolase is going to turn that acetoacetyl-CoA back into two acetyl-CoAs. That'll just split it right in half. And that acetyl-CoA can run the TCA cycle. And of course, that TCA cycle is how we get ATP. So that's how we're getting ATP out of our ketones. Back to the outline, same thing I just walked you through. If we made beta-hydroxybutyrate, we're going to turn it back into acetoacetate via that beta-hydroxybutyrate dehydrogenase. Then we use that new enzyme thiophorase to take acetoacetate and combine it with succinyl-CoA from the TCA cycle. And that gave us our acetoacetyl-CoA. Then we split that acetoacetyl-CoA apart via that enzyme thiolase into two acetyl-CoAs, and those acetyl-CoAs were good to go run the TCA cycle. And that TCA cycle gave us ATP. All right, so let's wrap things up. Ketones are what we were talking about in this video. They're our water-soluble energy source released during prolonged glucose deficiency. And the main situations we talked about where that is the case was starvation, sort of late-stage starvation, diabetic ketoacidosis, or alcoholic ketoacidosis. We took acetyl-CoA, we put it through a series of reactions and turned it into acetoacetate to start, which is sort of our base ketone. And from there, we could turn it into beta-hydroxybutyrate or acetone. Acetone gets exhaled into the breath, giving people a fruity odor. Acetoacetate and beta-hydroxybutyrate could go out to peripheral tissues, to their mitochondria, to be used for energy. They get broken down basically into their acetyl-CoA constituent components, which can go on and run the TCA cycle. And that's how we get energy from those ketones. We talked about ketoacidosis, which is the state of having too many keto acids in your blood. In essence, the inciting event between DKA, starvation, and alcoholic ketoacidosis is a depletion of oxaloacetate. That causes a buildup of acetyl-CoA, and that acetyl-CoA diverts into the ketogenesis pathway. And we were thinking about ketoacidosis of one etiology or another when we saw things like polyuria, polydipsia, that fruity breath odor, nausea, vomiting, abdominal pain, and altered mental status in the context of a high anion gap metabolic acidosis. We're going to talk more about ketones in the next video, the last video for this chapter, which will talk about the progression of the fasting state into late-stage starvation. Till then, that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige, and thank you so much for joining me for our lesson on the fed versus the fasting state. So this is going to be a synthesis video of a lot of the different energy production processes we've been talking about over the last couple of chapters. You're already familiar with the fed state and the fasting state, right? The fed state is an anabolic state driven by insulin, while the fasting state, or as it progresses into the starvation state, is catabolic and driven by glucagon and epinephrine. And how we get our energy in that fasting state or that starvation state is going to change based on how far along that starvation process we are. So the relevant energy production pathways change as our available fuel sources deplete. Something to keep in mind as we move through this lesson is that the brain must use glucose or ketones for energy. Those are its only two options for an energy source. Even more restricted are red blood cells, which can only use glucose, and they don't respond to glucagon or insulin. So we always have to make sure we have glucose at minimum available. And that's going to dictate a lot of which of our energy production pathways are running at any given time. And different energy sources have different efficiencies as to how much energy they can actually give you. And every once in a while on the STEP exam, they'll give you a weird macronutrient calculating question. So I would actually commit these numbers to memory. Carbs and protein give you 4 kilocalories per gram. Alcohol gives you 7 kilocalories per gram, and fat gives you 9 kilocalories per gram. So let's start off by talking about exercise and the fuel sources that fuel our exercise effort. A full lesson on this topic can be found elsewhere on the site in the MSK unit on the metabolic exercise physiology video, but we're going to review it here from a biochemistry lens as well. So you can look at this first bullet point here on the outline as sort of like a timeline. The start of our exercise effort is sort of over here at time point 0, and then you can see how those exercise fuel sources change as you progress through the exercise effort. In the first burst of exercise, in the first couple seconds, we're going to have 2 energy sources. The first is stored ATP, and this will be released immediately. Remember, your body stores very little energy in the form of ATP itself. Instead, it holds a reservoir of phosphate bond energy in the form of creatine phosphate. And if you need a refresher on that, go ahead and check out the video on ATP export and phosphocreatine in the energy production chapter. So stored ATP and creatine phosphate were our energy sources in the initial seconds. That was the reservoir we already had ready to go. Once we use all of that up, our next line of defense is anaerobic metabolism, right? Anaerobic glycolysis. And different sources are going to give you different exact numbers for these durations of time. Less important than knowing the numbers themselves is going to be to understand the progression, how we move from the beginning of an effort to the end of an effort. And so we'll say for about the next 10 seconds, anaerobic glycolysis is really dominating. And the good thing about anaerobic glycolysis, as well as the phosphocreatine system, is that we can kick it into gear really quickly. However, the downside is that it is not as efficient as some other processes like aerobic metabolism in forming ATP. And that's because we're creating lactate as our end product instead of pyruvate. So I put together a compilation of a lot of the different pathways we're talking about here, just so you have them all in one place for reference. And if you check out our diagram on the left here on glycolysis and gluconeogenesis, anaerobic metabolism takes place outside of the mitochondria and forms lactate, as opposed to aerobic respiration, which will form pyruvate and enter the mitochondria and run through the TCA cycle. So you can see anaerobic metabolism, that pathway is much shorter, which means that we can access that energy more quickly. So it's good for early on in our exercise burst, but as we continue to progress and we need more and more energy, it's worth it to invest more in that longer pathway for a larger payout. So that anaerobic respiration is going to be limited by carbohydrate stores, so things like blood sugar, as well as glycogen in the liver and muscle. And so once we reach those limits, we're going to start transitioning from anaerobic metabolism to aerobic metabolism. And then after about 1 minute of consistent exercise, aerobic respiration is going to be what dominates. Remember, we said that this has a low rate, it's a longer pathway, but we get a higher ATP yield in the long run. So from 1 minute onwards, we're going to be running that aerobic respiration. As we continue on our exercise through minutes to hours, we will add on glycogenolysis and beta oxidation as auxiliary energy production pathways. Your peak overall performance is already on the way downhill by 10 seconds in and is remarkably decreased after about 30 seconds. All right, so those were our energy production resources as we progressed through an exercise effort. Now we're going to be walking through the different energy production pathways that are in play as we progress from a meal through the fasting state and eventually through starvation. So we can start up here at time point 0 of our last meal and progress through the stages of starvation. At time point 0, we're in the fed state or the postprandial state. Here we've got plenty of glucose in our blood, and we can use that to run glycolysis and aerobic respiration and even store away some of that excess as triglycerides. So this box right here represents the fed state, where we have plenty of glucose and we can go down and run aerobic respiration to form pyruvate, which can enter the TCA cycle for energy. And if we even have extra acetyl-CoA from running that pathway, we can run it up to the triglyceride synthesis pathway and store some of that energy away for when we need it later. After a couple hours following our meal, we will be in what we call the short-term fasting state, and that state is going to be dominated by glycogenolysis. So now we're looking up here at our glycogen metabolism pathway. Remember, we could break glycogen apart into intermediates that could then go down and run the TCA cycle in the absence of blood glucose. And the full details of that pathway are on this diagram here on your compilation slide. But those glycogen stores only last about 1 day. After 1 day until about 3 days after your last meal, you are in the early starvation stage. That's going to be dominated by 2 energy production pathways, lipolysis and gluconeogenesis. Remember, in the adipocytes, the muscle, and the liver, we can run lipolysis, which broke apart those storage triglycerides that we were talking about earlier into free fatty acids, favoring the use of free fatty acids over blood glucose. Those free fatty acids could be broken down into energy via beta oxidation, the byproduct of which was acetyl-CoA. On your summary slide here is the fatty acid metabolism pathway. And here was the pathway where we took fatty acids, brought them into the mitochondria, ran them through beta oxidation to eventually form acetyl-CoA, which could either join the TCA cycle or be used in ketone metabolism. And that's a great source of energy in our back pocket. But remember, because our red blood cells can only use glucose as an energy source, we need to be making glucose for them. So at the same time as all of this, the liver is also going to be undergoing gluconeogenesis. And there are lots of different starting substrates for gluconeogenesis, like we talked about in our gluconeogenesis video. We could use lactate or alanine from peripheral tissue, as well as glycerol and propionyl-CoA from odd-chain free fatty acids. So you can see our early starvation energy production box over here. So in beta oxidation, you can see how these fatty acids are broken down into acetyl-CoA, which can run the TCA cycle, as well as odd-chain fatty acids, which I wrote in down here, which can also join the TCA cycle. And we're going to kick up gluconeogenesis as well, making sure that not only do we have energy, but we actually have energy in the exact form of glucose for our brains and for our red blood cells. During the early starvation stage, your brain is going to use the glucose from gluconeogenesis, and your body can use those triglycerides. Remember, we have to have that gluconeogenesis going because we must have energy in the form of glucose for our red blood cells. And additionally, our brains can't use triglycerides. They have to use either glucose or ketones, and in the early starvation state, we're not kicking up ketones yet. Ketones will come into the picture in the late starvation stage, which is after about 3 days after your last meal. That is when ketosis will really start to dominate. Remember, beta oxidation producing acetyl-CoA can also feed into the ketone metabolism pathway. Here we are on our diagram. Here is late stage starvation, and you can see that that acetyl-CoA that we produced during beta oxidation, not only does it run the TCA cycle, but it can join the ketone metabolism pathway. And that's the pathway that's going to start to dominate in late stage starvation. However, gluconeogenesis is still going to be ongoing during this time. Remember, it's great that we have all of these other energy sources, but we must have a source of glucose for our red blood cells. Our brains can use the glucose or the ketones that we've made during this time, and your body can continue to use up triglycerides. So that's the late starvation stage. And that late starvation stage, you can see we just wrote greater than 3 days. That's going to keep going on basically until you run out of energy stores. No more energy stores means you cannot make ketones anymore. And your body, in one last ditch effort to make energy, is going to start degrading proteins in your body for gluconeogenesis. And eventually it will start to degrade your muscles and your organs. And that is when you reach organ failure and ultimately death. Here is our end stage starvation box. And there are a lot of different ways that different amino acids can enter the energy production pathway. Some we've talked about, some we haven't yet. We'll talk about these in our chapter on protein metabolism, but I've given you some different examples about how different amino acids can enter into this TCA cycle and feed into gluconeogenesis. And for a more complete picture, I did add a diagram on amino acid catabolism on your summary slide, and this is just a preview for you for now, but you can see some amino acids feed into gluconeogenesis, some feed into ketogenesis, and some can do both. And these different amino acids have different entry points into this energy production pathway that can ultimately go up and run gluconeogenesis. Obviously, though, you don't want to be degrading your organs until it is your last resort. So you can see here our diagram of stored energy on the y-axis versus the weeks of starvation. You can see almost immediately you run out of carbohydrate storage. Fat stores will carry you along much further until eventually they die out. And once those start to drop dangerously low, it is only then that protein catabolism kicks in as a last resort. So let's wrap this up. We've been talking about the fed versus the fasting state. We started off by talking about exercise fuel sources and the different sources of energy that we could use throughout an exercise effort. We started by releasing stored ATP, as well as our creatine phosphate stores, progressed on through anaerobic metabolism, then aerobic metabolism, and then eventually we could even add in glycogenolysis and beta oxidation as we got far enough down along through that effort. Then we took inventory of all of our different energy production pathways as we progressed from time point 0 with the intake of our meal through the stages of starvation. In the immediate postprandial state, we could run glycolysis and aerobic respiration and even synthesize triglyceride stores. After a few hours, we started relying on glycogenolysis, which were carbohydrate stores. Those carbohydrate stores only lasted about 1 day. And then we started relying on lipolysis and gluconeogenesis to get us through the early starvation state. Once we reached the late starvation state around 3 days, we started to rely very heavily on ketones. And we kept burning through those ketones until eventually our energy stores in our body completely ran out. And that time period really just depends on how many stores you were starting out with. Once we reached that end stage starvation at the end of our energy stores, we started degrading protein, which eventually led to organ failure and ultimate death. And we'll talk about protein metabolism all in our upcoming chapter, so stay tuned for that. Till then, that's all I got for you for this chapter. If you have any questions, comments, concerns, problems, troubles, worries, hopes, dreams, fears, send them all my way. Otherwise, good job, good luck, and keep practicing. All right, go ahead, take a minute, read through the question, and then we'll come back to talk about the answer. So we have a 57-year-old woman who is presenting to the emergency department with a 2-day history of intermittent right upper quadrant pain, nausea, and bloating. The pain is worse after meals and is radiating to her right shoulder, and she's had similar episodes in the past. Her past medical history includes type 2 diabetes, hypertriglyceridemia, and hypertension, for which she is taking metformin, fenofibrate, amlodipine, and lisinopril. Her vital signs are within normal limits, and her physical exam is revealing some right upper quadrant tenderness. So they went ahead and did a point of care ultrasound, and they're going to show it to us over here, which we'll walk through in a minute. But first, let's walk through her labs. So the white count looks normal, the bilirubins both look normal, the LFTs look normal, and so does the alk phos. So she's got normal vitals and normal labs. Great. So the question wants to know which of the following is the most likely pathophysiologic mechanism underlying the patient's condition. So this patient is having intermittent right upper quadrant pain, worse after meals, radiating to the right shoulder. She's had it before. This is a classic story for biliary colic. Her vitals are within normal limits, so she has no fever. Her white blood cell count is fine. She has no sign of infection. Her liver function tests are normal, so there's no evidence of like a damaging obstructive process. So we're just looking at biliary colic, which is an uncomplicated cholelithiasis picture, or gallstones, without any sort of cholecystitis or choledocholithiasis or cholangitis, right? Those are infectious and inflammatory processes that would drive up some of those labs. So we're thinking biliary colic, we're thinking gallstones. Let's take a look at the ultrasound. And you don't have to be a master at point of care ultrasound to interpret some of these basic ones. We know we've got a right upper quadrant ultrasound, so we can intuit that this large anechoic, meaning fluid-filled, structure in the middle is our gallbladder that's full of bile. And then we've got these hyperechoic, or bright, balls basically sitting there in the gallbladder. And there's this posterior shadowing behind it, right? You see how it kind of looks like these stones are casting a shadow back behind here? That posterior shadowing means that there's something super dense sitting in front of the shadow. And it's dense enough that the ultrasound waves that are coming down can't penetrate through. So these are gallstones right here, and I bet if we had a video and if we asked the patient to roll around, we could see those gallstones moving around. They're gravity dependent. If you didn't know how to read the ultrasound though, there's enough information in the stem to answer it without the ultrasound at all. This woman has a lot of risk factors for gallstones, and the mnemonic that people like to use to help remember the risk factors for gallstones is fat, female, fertile, and 40 plus, referring to age. And while we don't have all the details about this patient's BMI or pregnancy history, we do know that she is a woman over the age of 40. And I'll explain why these things are risk factors in a minute, but there's one more risk factor that I want to make sure that we see. This patient is on a fibrate, fenofibrate. And fibrates have a famous side effect of causing gallstones. So let's talk about how each risk factor works, and that will help us answer the question. All right, so let's remember, what are the components of bile? Bile is made up of bile salts, cholesterol, and then some other things like phospholipids, bilirubin, water, proteins, things like that. For our discussion, we're focused on bile salts and cholesterol. Bile salts are like the dish soap that keeps the cholesterol grease from clumping together. They help to emulsify the cholesterol so that they dissolve out into the bile. And a bile salt is a bile acid that is conjugated to something like glycine or taurine so that it dissolves more readily. So like I said, bile salts are like our dish detergent that helps keep the cholesterol dissolved. So our risk factors for gallstones are either going to be things that decrease the amount of bile salts that we have available to emulsify and dissolve the cholesterol in the bile, or things that increase the amount of cholesterol in the bile so much that it overpowers all those bile salts. Both of those downstream will lead to gallstones. So each of the risk factors in this question can be attributed to one or both of those underlying causes, either a decrease in the production of bile acids and bile salts, or an increase in the concentration of cholesterol in the bile. So each of our risk factors in this question can be explained by one or both of the underlying risk factors we just established, a decrease in the production of bile acids and bile salts, or an increase in the saturation of cholesterol in the bile. Female sex is a risk factor because estrogen upregulates the activity of HMG CoA reductase. HMG CoA reductase is an enzyme in the cholesterol synthesis pathway. Increased cholesterol synthesis, obviously, is one of our risk factors. Age over the age of 40 is another risk factor because as you age, the speed at which you produce new bile acids and bile salts decreases. Fibrates are a risk factor for both reasons. They both increase the amount of cholesterol and decrease the amount of bile acids. Here was our diagram of hyperlipidemia pharmacology, and here were fibrates. And the way fibrates work is they activate this right here, PPAR alpha, which upregulates the activity of lipoprotein lipase, or LPL. LPL enhances triglyceride breakdown, and this will end up mobilizing more cholesterol into the bile. So that's how we end up with cholesterol supersaturation in the bile. But the other thing that fibrates will do is they will inhibit the activity of 7-alpha hydroxylase, which is the rate-limiting step in the bile acid synthesis pathway. So you can see why fibrates are a big risk factor for gallstone production, as they both increase the amount of cholesterol in the bile and then reduce the number of bile acids you have available to solubilize it. With this in mind, our best answer choice is A, decreased bile acid synthesis, although increased cholesterol saturation in bile could also be an acceptable answer choice if we had it here. Let's walk through our incorrect answer choices. Answer choice B says increased enterohepatic recycling of bile salts. So enterohepatic recycling, entero means intestine, hepatic meaning liver. This is referring to the phenomenon that a large percentage of bile acids can be reabsorbed from the ileum and go back to the liver. So those bile salts made in the liver, they go to the gallbladder, they get excreted out into the duodenum, and they follow the small intestine around till they get to the ileum, at which point most of them actually get reabsorbed and go back to the beginning. That allows us to conserve bile acids, and then a small amount will continue on to get fully excreted. Increased enterohepatic recycling of bile acids would be a good thing and would be protective against gallstones. So this is not correct. Our next answer choice increased unconjugated bilirubin precipitation. This is representing a pigment gallstone. These are the black gallstones that aren't caused by cholesterol. They're instead caused by a bunch of increased unconjugated bilirubin, often in chronic hemolytic diseases. We have no reason to believe that our patient is chronically hemolyzing. Pigment gallstones in general are less common than cholesterol gallstones. Plus, this patient's bilies are totally normal, so this is not our answer. Our next incorrect answer choice is impaired gallbladder motility. And yes, gallbladder stasis can contribute to gallstones. When the gallbladder is very still, all of that dissolved cholesterol can precipitate out of solution. So yes, this is a theoretical correct mechanism for gallstones. Problem is this patient has no risk factors for gallbladder hypomotility. Our risk factors for biliary stasis are pregnancy, fasting, critical illness, you'll see this a lot, and somatostatin analog medications. The risk factors for gallstones that our patient has don't cause gallstones via that mechanism. So that is incorrect. And then our last incorrect answer choice is increased biliary excretion of triglyceride-rich lipoproteins. Lipoproteins, remember, are things like chylomicrons and VLDLs. These were our lipoproteins. They were the way that we packaged triglycerides and cholesterol and cholesterol esters in the lipid absorption pathway. These lipoproteins are not excreted into bile. Yes, fibrates promote the breakdown of triglycerides, but those triglycerides don't get packaged into lipoproteins and sent into the bile. We already talked about our components of bile. There are no lipoproteins in there. The cholesterol that is excreted in bile is free cholesterol. It's not packaged into lipoproteins in the same way it was when it's absorbed. So choice E is out. So in summary, we had a case of biliary colic, which is an uncomplicated case of gallstones in the gallbladder, which causes pain when the gallbladder contracts around the stones. But unlike some other underlying pathologies, labs are normal, there's no underlying infection or real dangerous or pathologic blockage. The risk factors for gallstones that you can remember are fat, female, fertile, 40 plus in age, and then I'll give you a fifth F, which is fibrates. Those risk factors mainly act by increasing the amount of saturation of cholesterol in the bile and decreasing the amount of bile salts that are available to solubilize that cholesterol. Fibrates in particular interfere with bile acid synthesis by inhibiting the activity of 7-alpha hydroxylase. And that's all I got for you. Good job, good luck, and keep practicing.